No, that she was presenting, and suddenly it's like a little circle appears, and there's like there's some audio as well, but yeah, then quickly. I think the problem is
Good morning, Sharon.
so you're listening to Evelyn already?
This Dromika is absolutely fascinating South African bidders. I suspect some of you know about them. Very fearless, flightless cicendelids, which, as you can see from those mandibles, are very aggressive predators of other insects. Anyways, I had a fantastic trip and fantastic memories, and I really would like to visit you again one of these days. But this is the South African Society for Plant Pathology, not the South African Society for Entomology. So I'll be going back to my theme today, which is plant pathology. And of course, this year we are celebrating the International Year for Plant Health. This is, as you know, a very important topic for our field, plant pathology. Unfortunately, it was hijacked by another type of pathogen, but nonetheless, I think this is an opportunity for us to reflect on some of the bigger questions related to plant health and plant pathology. So this, again, will not come as a surprise to many of you. Plant diseases have increased in frequency in the last years. Increased trade, meaning introduction of new pathogens. Climate change also is creating a lot of pressure on our crops. And of course, these pathogens evolved, and occasionally they will jump from one host to another, in a way similar to what we're seeing with that dreadful coronavirus. The same thing is happening with plant pathogen, and occasionally you do have new pathogens emerging on crops because they move on from wild plant populations. Nonetheless, despite all these new outbreaks, I have a feeling that plant pathology occasionally is one step behind in terms of diagnosing and really calling out these new outbreaks, especially using the latest technology like genomics technology. So this is taken from ProMed Mail, which is the one organized system that really reports based on literature survey and news announcements, that reports new outbreaks of plant diseases. And if you do like what my colleague Ksenia Krasileva did, is make a word cloud out of all these ProMed Mail reports, then you'll see that the word undiagnosed is actually the most common one. And that's really sad, because we're really at the stage where with genomics, with genetic analysis, it's fairly straightforward to actually diagnose these pathogens almost in a large scale, high throughput fashion. And it really saddens me that many of these diseases, most of them are in developing countries, are really not being addressed using this newest technology. So this is where I start actually in this story, because back in 2012, I was looking at what's being done with human and animal diseases, and I was really feeling quite sad actually, and quite annoyed by the fact that plant pathology hasn't embraced genomics surveillance, genomics diagnostics in the same way as our colleagues working with biomedical problems or even with foodborne diseases have been dealing with new outbreaks. So this is really the starting point for me. So back in 2012, I wrote this commentary, which was really arising out of frustration, if you like. And then I thought, okay, I really want to showcase how we can apply genomics technology to a new problem. So I told my lab, look, as soon as we hear of a new problem, we should really apply our expertise to address that problem using genomics, and showcase how genomics can really impact this. And an inspiration, which is something that is obviously very relevant today, is this platform called NextStrain. It's a wonderful platform. It was originally developed for the flu, for influenza virus, and of course recently was applied to the SARS-CoV-2, the coronavirus. And this is essentially a repository of all the genomics data being done all over the world, UK, South Africa, everywhere else. And every time a new coronavirus strain is sequenced, it's actually deposited and cataloged in this platform, in this next strain platform. And I encourage you to visit the site. It's very informative if you want to learn about the virus evolution and virus diversity. It's a very interesting website. It allows them also to have this sort of interactive features about where particular lineages of this virus, how they're moving, where the polymorphisms are, and these sort of things. But what's really interesting about this, and really again, this is the inspiration for a lot of the things I do, is that NextStrain is, first of all, an open source project. All the data is publicly available. All of the data is openly shared. You can go download data, play with it, analyze it, add your own data. It's really all open. The second thing, NextStrain generally doesn't generate its own data. So they're primarily actually curating and sharing public data, organizing the data that is out there. And finally, there's a huge investment there in data visualization and analysis. So that aspect actually is also very important. How do you share the data and turning all that raw data into something that is easy to use and process. So to cut a long story short, and really to sort of, we have moved forward with this idea of genomic surveillance of plant pathogen, and it all culminated into this exercise where a group of us met in Bellagio in Italy a few years ago, a couple of years ago now, led by Monica Carvajal at CIAT, and came up with this policy forum and this concept of a genomics or a global surveillance system, GSS, if you like, for crop diseases. Things have been slow to develop beyond that, mainly gaining funding and all that. But the idea in terms of genomics is really that we need a lot more data. We need 1,000 times more data than we have today. This is very important. Again, I think we have now this current crisis with the coronavirus as a good example where having more data, it is actually useful and provide useful knowledge. Also, speed is critical. Speed is really essential. We have to move fast to address these problems, especially when there are new outbreaks. We can do genomics directly from the field. We don't need to go through the tedious process of culturing, purifying strains, etc. We need to integrate genomics with other type of data that plant pathologists collect. And there are wonderful platforms out there in terms of collecting phenotypic data, collecting data straight from farmers and all that, but that needs to be integrated also with the genomics data. And finally, the importance of open source, and the consequence of open source really is crowdsourcing, in the sense that now, once the data is open, anybody who has expertise and knowledge can actually contribute to the analysis. So this is where we are today, but what I'm going to do now is really take you through what happened between essentially 2002 and 2009, 2012 and 2019, sorry, I lost track there. And since essentially that kind of aha moment I had when I thought like, we really need to apply genomics to these problems versus that GSS concept, what happened is we had a few emergencies that we decided to address. And we decided to address them in a different way. It was very evident that we need a new way of doing business. The old way of doing business, the classical way, it's a very stratified approach. There's someone at the top, generally an organization, maybe CGIAR center or something like that, essentially assigning people different tasks. And this is done in a very structured way, which is not the optimal way to address an emergency. What we need is essentially what's on the right side there of that cartoon is a more interactive way, a little bit of a mess sometimes, but that's fine. It's okay if multiple people do similar things, but this allows to move in the fastest way in addressing the emergency. Anyways, I'll show you how this works. So the first problem we faced was in 2013 was a tree problem, actually, an ash tree. Problem. So ash dieback is the disease. It popped up in the UK for the first time in November 2013. It was very destructive. Interestingly, this type of problem gains media attention. As you can see there, it was on the cover of the Times magazine, even got more space on the cover than the new James Bond movie. So that's good for plant pathology. Hey, plant health is important, right? And as you can see there, it was really threat to the 80 million ash trees in the UK. When we heard about that, that was the first case where we said, okay, let's kind of get involved with this. And we essentially, within a month, we released the genome sequence of the pathogen that helped identify its source, how it was introduced to the country. We also helped us realize that it is a sexually reproducing pathogen, very diverse, which resulted in a different way, really, of dealing with the problem, and also in terms of selecting trees that are resistant and all that. My colleague, Dan McLean, who is shown there, went on even to involve the public in a sort of citizen science type of approach, where he actually developed a game where the citizen scientist could play this game on Facebook, which was solving a puzzle, which actually allowed us to improve the calling of mutations and polymorphism in this project. He published all of this. If you're interested, I'm happy to send you more information. And actually, the players can improve on existing computational programs. So that was an interesting side thing from that. But the approach we used is interesting. So the methodology was really doing RNA sequencing, RNA-seq, on infected tissue. Okay, so we don't need, this is why we could move really fast, is we didn't go initially through the process of purifying, culturing the fungus, etc. We just took the tissue, sequenced it, and showed that that information we get is actually very useful. First, in identifying which are the tree sequences and which are the fungal sequences. So you can see that cloud in the lower right there, the infected tissue. In the infected tissue, you could really clearly separate the sequences from the fungus from compared to the plant sequences. And that's enough to give you a lot of information enabling very precise identification of this fungus. So we call this method field pathogenomics as a general way of basically generating genomics data from infected tissue collected directly from the field without going through the process of culturing and all that. Obviously, this is really critical for non-culturable organisms. Then you can really move really fast. You don't actually have much option, but it is useful for any kind of system. It's a very fast pipeline to get genomics data. Okay, so now fast forward to 2016. So in 2016, we came across a new outbreak in Bangladesh. So these were the first reports that there was a new disease on wheat that hasn't been reported before. It looked like wheat blast, but its origin, the precise identity of that strain was not clear. Whether it was an endemic strain that jumped to wheat or whether it was introduced, that really wasn't clear. It was very destructive. 15,000, 16,000 hectares were destroyed in a short period of a month and a half or so. So we used social media then to actually connect with our Bangladeshi colleagues. So Tofazal Islam was someone I met at plant pathology congresses a couple of times. We became friends on Facebook. I knew of him through the Facebook connection. I knew he was quite a dynamic fella over there in Bangladesh. So I immediately sent him a message on March 8th, actually, 2016, asking him, well, can you get samples? Can we apply this method we have to this problem that arose in your country? And he immediately went very active, actually used social media, and a lot of the extension people there have smartphone, they're on Facebook. So he could immediately really get into the field and get us samples directly from the field. So that really happened very fast. And we decided to do the sequencing as fast as we can and put all the data out there on this platform we called Open Wheat Blast. So Open Wheat Blast was our portal, which, without doing any analysis, actually at that time I had no idea about wheat blast. I really didn't work at all with this pathogen. So we really needed input from the community. So we posted the data. There was a lot of media attention that came through that on the cover of Bangladeshi newspapers all the way to international scientific magazines. And what was fascinating is within weeks, really, we had people contributing analysis of the population analysis of this outbreak. So we had two actually independent analyses, one by Daniel Kroll in Switzerland, and another one by Pierre Gladieux in France. And this really happened essentially 6 weeks. They posted their reports 6 weeks after field collection of the samples. So within 6 weeks, we went from field collection in Bangladesh all the way to going live with the website to having the community contribute reports. What was really fascinating to me is we needed the community input because there was actually only one genome sequence of a wheat blast strain that was public when we went live on April 18th. But then many members of the community said, we have this genome sequence, they're sitting on our servers, we haven't published them, we want now to contribute them to help analyze data. At the end, we had something like 25 strains or something, 25 different genomes contributed by different labs. But essentially, nobody could really do the analysis on their own without the community coming together and sharing these resources. To cut a long story short, it turned out that the isolate was very likely introduced from South America, which is not totally surprising given that Bangladesh imported some suspiciously looking wheat, let's call it like that, from Brazil just months before the outbreak. So that's probably how the pathogen was introduced into the country. And again, with the timeline, you can see here that we posted the preprint in bioRxiv in June 19th. So literally 6 months after the samples were collected from the field in Bangladesh. And then we formally published the paper in a journal, in a peer-reviewed journal in September. So essentially 9 months after the samples were collected. So I thought this was a really good showcase of how we could rapidly rally the community to together address a problem like that. You can see we had multiple authors, including folks from Brazil who also had their own data to contribute, etc. And again, the approach, field pathogenomics, we could easily, by sequencing RNA from infected lesions and both symptomatic and non-asymptomatic tissue, we could separate fungal sequences from the wheat sequences, and the fungal ones are shown in blue there. So it was a little computational trick that allowed us to go really fast in analyzing the sequences and not delay it by culturing and all of that. So, okay, this is the data. It's not really important, but the point is that this Bangladeshi population turned out to be a clonal lineage, single clone. We have been following it over the years. It's just a single clone that is actually diversifying at its rate, but it is out of the South American stock and very closely related to South American isolates. To kind of summarize what we did there, what we learned really is, first of all, we learned that genomic surveillance can be rapidly applied to monitor a new plant outbreak. And we can generate very valuable information regarding the precise identity and origin of the infectious agent. For example, the knowledge that we acquired in South America, the wheat blast emerged in South America in the 1980s, that knowledge could be immediately applied to Bangladesh, fungicides, cultivars, etc., that could be used. And immediately, CIMMYT actually established a field trial that was done in parallel with exactly the same material, both in South America and in Bangladesh, as a way of testing different varieties and how they perform. But the fact that we knew that it was similar populations of the pathogen was really important for arguing for that investment. We also could identify through genomics the effectors, the virulence effectors, for example, and therefore guide which R genes can be used. So one gene that came out of that is RMG8. It's a gene that was not effective against most of the Brazilian populations of wheat blast, but the AVR effector is present in the Bangladeshi clone, and therefore RMG8 is effective against the Bangladeshi population. And that was an argument through sequencing to tell our Bangladeshi colleagues to consider using that gene, and they are indeed testing cultivars with RMG8. And finally, the crowdsourcing. I mean, there's many aspects of this, not just getting people in the world who are really the experts in this problem to contribute, but also bringing the community together. I think working together, get everyone to know each other, establish new connections, new links, and so on, and really help set the stage for many follow-up analysis, which actually did happen in many cases, exchanges of visit, of technology and knowledge, etc. So what are we doing these days? As I said, we're following the Bangladeshi clone over the years. It's now 4 seasons, and we have 4 seasons of data in terms of genomics analysis. I don't really have much time to talk about that, but one question that was really of concern from day one was whether this pathogen newly introduced to Bangladesh in the greenhouse had a fairly wide host range, whether it was actually going to jump to other crop plants in particular, but maybe even to other grass hosts, and that would affect also the epidemiology. And Bangladesh, unlike Brazil, most of the field plots are actually quite small. We have typically small-scale farmers, and as you can see here, for example, maize, which is actually susceptible to the blast fungus, for example, is right next to a totally infected field of wheat, and also rice and wheat are often grown side by side. So we have an endemic population of rice blast. We have a new conspecific lineage wheat blast introduced to the country. What's the potential for host jumps? What's the potential also for exchange of genetic material? Fortunately, the suspicious lesions that were observed on maize turned out not to be wheat blast. So far, a jump to maize hasn't occurred in the field, at least. But to monitor this, we develop a variation on our method, which is based on amplicon sequencing. And this is really a brilliant method developed by my colleague Kurt Lamour in University of Tennessee. And he has actually a small company, Floodlight Genomics, applying this methodology. And essentially, it's a one-tube PCR where you mix together a lot of primer pairs, in his case, 85 primer pairs, that amplify different regions of the genome and reveal the sequences of individual polymorphisms that are actually diagnostic for the population you're studying. So it's a very quick way. And then what you do is when you have all these multiple PCRs, they're just actually pulled together and sequenced by Illumina type sequencing in one go. So it's they are barcoded and then sequenced, and then there's some computational work to extract the information afterwards. It's a very quick and cheap way of actually establishing surveillance. And we showed that we can do this from, again, lesions collected in the field, where you just extract DNA, in this case, from the lesion and run it through the PCR. And that's good enough to give you data that allows you to separate, in this case, the BD clone shown on the left side, that arrow there with BD. So those are actually identical. They're all the same clone, different isolates from Bangladesh, from wheat. And then we can really distinguish that clone from other South American wheat blast and any other active blast fungus, including local endemic rice blast isolates from Bangladesh. So this allows to see if there is a jump, a host jump, if the BD clone would move to a new crop, a new grass. Any suspicious lesion can be sequenced, analyzed, and also it will allow us to see whether rice blast can also move to other strains and so on. So it's a good way to monitor essentially your population in a large-scale fashion. The methodology is very easy. Essentially, as I mentioned, we just collect lesions. Essentially, it's collecting herbarium type material. You basically collect lesions, dry them up, send them over to the lab, and then DNA is extracted and run through Kurt's pipeline. This is somewhat relevant to you in South Africa now, because what happened is there was a report just last September 2020, that there is a new outbreak of wheat blast in Africa for the first time in Zambia. And this is the scientist, Dr. Batiseba Tambo, looking at the totally destroyed, really, wheat field with the typical wheat blast symptoms. It's really tragic that the disease has now made it to Zambia, to Africa. I was told that Zambia actually does export wheat to neighboring countries, although so far the reports are only of the rain-fed grown wheat, so the small-scale farmers rather than the larger farms that are actually the ones that typically export out of the country. The report only came in September. We actually didn't know about it, but one thing that was a bit puzzling to many of us, especially because there were many international scientists involved in the publication, is that although nobody knew about this up to September 2020, the first outbreak was in the 2017-18 rainy season, field season of growing wheat. So basically the pathogen popped up 3 years ago, and then it took quite a while before this was actually made public, and it was first announced through a formal publication, plus one, as you can see, a formal journal publication. So I would add that since the report came, we're now involved with our Zambian colleagues and CIMID colleagues to analyze this population in Zambia. So we just received actually isolates from them, from Dr. Batiseba just last week. So we'll be hopefully making data public very soon as we have sequences available. Same approach will be all through Open Wheat Blast. But this is a good transition to me to the topic now, the last part of my talk, which is not really about science, but rather how we share our scientific knowledge. How should we really act during emergencies, plant health emergencies? And I think the Zambian example is sadly not a very good one in how we should approach these problems. The problem is there, it's being done in a very traditional way. It's done through scientific publications in peer-reviewed journals. Now, there's nothing wrong with that. But when you have an emergency, you can't just wait for that. You can just wait for several years until you have a fully developed paper that has gone through peer review, and sometimes with all the delays that it takes before actually you make the announcement that you have an outbreak. We really have to deal with plant health emergency differently. Many challenges, as you can see in this slide. Funding is an issue, collaboration, crowdsourcing versus a top-down maybe, or an individual type of collaboration. Open science, immediate release of the data versus journal publication. And I will discuss a little bit more about this publication concept because that's also changing a lot now. We have new forms of publishing like preprints and new ways of actually really getting the data out there, while at the same time the scientists who develop that still get the credit that they deserve. And then finally, there's the issue of research assessment. Currently, a lot of research assessment does not actually acknowledge the diverse contributions of scientists beyond journals, and there's this unhealthy obsession with metrics. So let's talk about the new models and new ways of communicating research. So we have a lot of new platforms. We have preprints like bioRxiv is now the go-to preprint server for biology. We have post-publication peer review, in the idea that you actually post your paper and then it gets reviewed after it's being posted, after it's being shared with the community as a preprint, not before it is actually published. And then we have also new platforms like Zenodo, which are essentially repositories for data, preprints, posters, anything you like. So these new forms of publication, they're summarized here. We have, again, the preprints. Preprints tend to be fully fledged papers, but in many cases, we want to have actually shorter reports. For example, here, just reporting that you have sequences of certain isolates. You don't have time to analyze it or produce a full paper. In that case, these mini papers are appropriate for servers like Zenodo, which are essentially servers that allow scientists to share data in any way they like, but without it necessarily being complete for a fully developed paper. So it's a stage between your kind of lab report and a full publication. The key to all these is the fact that there is a DOI, a digital object identifier. What the DOI means that it becomes a citable unit, because the DOI means once you post it, you cannot modify it. It will be there for posterity, and it will be assigned a DOI identifier, which means that it is for most journals, for most publications, it's something you can now cite. You can use it to stake priority, etc. So for example, our little report there about nanopore sequencing of magnapore theorized isolates is a report that is permanently there as a Zenodo technical note that anyone can cite, and actually it's been cited in journal, etc. But what's really important to understand is that this does not prevent you from publishing in traditional journals. Once you post these reports, preprints or mini papers, etc., you can still develop them into papers that you submit to journals. Almost all journals allow the publication of preprints and pre-reports prior to submission to a peer-reviewed journal. So again, the concept here is publish and filter, not filter and publish. So that really turns the whole idea of publishing on its head. And it's obviously very rational and logical in the internet age where we now can share information as fast as we can. And obviously, this is extremely relevant for plant health emergencies. Very briefly, again, to explain this, I suppose some of you are not familiar with preprints, but just to explain it, with the traditional method, you submit the journal, there's editor, anonymous reviewers who would analyze it, and then it gets published. Now, with preprints, you just post it as a preprint. It's immediately out there for anyone to read, anyone to comment on. Sometimes these preprints get reviewed. Some of your colleagues may send you comments, etc. But at the same time, you can also submit it to a journal and going through the traditional process in parallel, in addition to your paper being shared as a preprint. So essentially, it's faster way of communicating your science to the community and not waiting for the delays that peer review causes. So for example, the Zambian outbreak really, which was published in PLOS ONE, should have been shared a year earlier. I was told it took a year for it to be published, could have been shared a year earlier as a preprint, and that would have alerted the community that there is indeed a wheat blast outbreak in Zambia, not waiting for this really slow, tedious process of peer review before it's actually being shared. Well, preprints are becoming very popular. They're very common in other fields of science like physics and math, but in biology, it's fairly new. bioRxiv was created, in 2013, 2014. As you can see here, it's becoming super popular, and that this is even outdated now. I guess the current numbers are probably off scale. Also, with the COVID situation, we saw the impact of preprints. There was a surge really in preprint papers. Almost everything that's being published on COVID is actually first posted as a preprint. So if you want really an example of how preprints can really accelerate the communication and the exchange of information, then look at what's going on with COVID. There was a number of articles even dating back as far as last May that discuss this preprint surge associated with the coronavirus outbreak. If you're interested in the topic, I highly recommend ASAP Bio, As Soon as Possible Bio. It's a very good name to describe this movement, the preprint movement. They have an excellent FAQ, which will address any of the questions or any of the concerns you have. So if you really want to know more about preprints, just visit this website. I told you about also other forms of publishing beyond preprints, mini papers, posters, etc. Like, for example, in my lab right now, we are regularly publishing our posters in Zenodo, which means that your posters get a DOI. They are actually citable units. There is an author list that is formal. Students can actually claim them in their CV and so on. So it's really good intermediate stage to make it more formal that the information you're sharing is actually recognized prior to actually becoming a full-fledged paper. And again, like especially for our work with genome projects, we first publish a report describing the data before we actually publish it publish the analysis. So that's what we've been doing with the BLAST project. For example, we created a community on Zenodo. You can actually create a community to collate and collect all these data sets. Sometimes people post data sets, sometimes they post videos, images. So this community is called Open Rise BLAST, but you can imagine all kinds of communities growing. For example, I can imagine a South African plant pathology community on Zenodo where reports, posters, etc., are being published in association with your own scientific community. And again, DOI is very important to understand this because we all care about credit, and of course, scientists need credit to move on with their career, to apply for jobs and all that. So the DOI is a very important part of this thinking because that essentially make your poster, your mini paper, your preprint a formal publication that you can actually refer to and cite, and that's really, really important. So I wanted to really be a little bit thought-provoking here and really share these ideas I have. Trying to be an activist for this new way of doing business, especially in relation to new emergencies. I think that's really, really important. But at the end, we all have to make it happen, especially in terms of rewarding and recognizing scientists. We really have to move beyond this traditional view that we should use journals as proxy for the quality of the science that is being produced. Rather, we should really analyze and evaluate the science itself independently of where it's being published. I can imagine how a report on a new outbreak in Zenodo as a preprint or as a mini paper would have a lot more impact than a formal journal publication that comes only 2 years later. So you can make it happen, and I hope to see more of these type of outputs from your community. So thank you very much. This is my team. We have different projects. I didn't talk about the NLR, the R-gene work we do on immune receptors. Some of that is on the internet, so if you're interested, you can check those talks. I have a group working on the BLAST disease, that's BLASTOff team, and then Adeline and Joe help everyone. And these are my colleagues at the Sainsbury Lab, really wonderful place to work. So that's all I have for today. I'm not sure if I was on time or not, but hopefully there's time for questions, and I suspect I'll be reading your questions in the chat. So so see you soon. Thank you very much.
Okay, thank you very much for that talk. It was just beautiful to listen to you. Certainly in time, and there is time for questions, but it was really enjoyable listening. It could have gone on for even longer. You've left us with much to think about, everything from how we are thinking about and managing plant pandemics, plant health pandemics. And that certainly is, I know, a topic that many of us share with you, being frustrated with 1, 2, 4 year delays on the announcement of the arrival of a new pathogen in an area, well beyond the time necessary to maybe reduce some of its spread or address that. The issue of wheat blast in Africa that you mentioned, but so much more in terms of how we can connect our global community, because sitting through looking at the things you're presenting and reflecting on how there's lack of capacity to do that kind of thing in certain parts of the world. But certainly if we can connect it in the ways that you've demonstrated, then we can address these issues. To the thoughts you shared about the broader open science movement. So thank you very much for a very rich talk, and I look forward to discussing that with you or with the rest of the community a bit further now. I see there's already discussions in the chat box. I haven't kept my eye on that. But ask Tuan, can people unmute themselves? So if you do want to ask a question, please just put up your hand or unmute yourself and make me aware that I'm not noticing you. So Fian, perhaps if I can get us started with this wheat blast outbreak in Zambia. You mentioned the fact that they export seed. And this is a pathogen that can spread through seed, I understand. Is there any effort to map its potential spread in other parts of Africa?
Thank you first. Thanks a lot for the great comments. I think that's really the idea here is, I think open science first, just going back to the first comments you made, I think one aspect of open science that sometimes overlooked is that it does bring people together, actually, these kind of open communities, because it's actually much better when you have this kind of naturally organic type of interactions developing based on common interest and based also on motivation, rather than a more of a formal top-down type of approach. So I think open science does foster that sort of spirit of creating networks that are organically forming and perhaps more optimal than structured organization and networks. But back to the Zambian problem, it was actually surprised to me. I was not aware that Zambia exports wheat, and I don't have a lot of details on that. I heard that through my colleagues at CIMIT, but definitely the strategy we are using of doing the amplicon sequencing from disease lesions collected from the field and just dried material, basically, we can process hundreds, if not thousands, of such material. So I would totally encourage anyone in this community and elsewhere, if you ever see anything suspicious that you think might be wheat blast on your crops, then by all means do get in touch with us and do collect those lesions. We can process literally dozens, if not hundreds, of these through the amplicon sequencing and immediately tell whether it's the wheat blast strain or not. In addition to that, I expect that we will be able to, once we have the genome sequence of the Zambian isolates, we'll be able to determine the origin of this pathogen, whether it's an endemic host jump or whether it was introduced somehow to the country. And of course, we can follow with great detail its spread. Really, it's not very different from what you're seeing with COVID situation, where it's actually possible to follow the spread of the pathogen in much detail, especially if it turns out that this is a clonal population, as we have in Bangladesh. It's clonal, so it's actually really fairly straightforward to follow over time. Obviously, it's not like with the virus, where it's a much smaller genome and easier to process hundreds of samples, but we have ways around that. So yes, it would be possible to follow the pathogen. On the other hand, I guess the fact that you can have such a detailed history of the spread is perhaps why there's reluctance to sometimes be very open about these problems. We have to be also accept that this is the other side, that there are consequences in terms of trade and in terms of limitations. And we've seen that play with the coronavirus too, right? I mean, to some degree, the UK and South Africa were pioneering in being first in reporting new variants of the virus, and it does seem like those two countries suffered perhaps more than the ones who were not doing any genomic surveillance. So this is also an issue we need to think about.
I guess with growing, such databases growing, it might get harder and harder to hide and lie about the origin and spread of these things. There are many more questions, and I'm sure others, but I'm going to jump to the chat line, Sophie, and address some of the questions there, or ask some of the questions. Brenda is asking there, I'm concerned about the potential of people publishing what is essentially fake data. How does one protect from this? Peer review does allow us to remove a lot of bad manuscripts out of the system. Do you have a response to that?
There's a lot of discussion about this, and I think going back to this tragic COVID situation, but I don't know if you heard the number, but there's been now 12,000 preprints posted on COVID in the last year. One very early on, a very kind of sensationalist preprint that came out very early, I think it was February, claimed that the coronavirus, the SARS-CoV-2, had a piece of HIV in it. And so they claim it was an engineered virus where someone in the lab spliced in a piece of HIV into this virus. And that was posted as a preprint, and that was immediately debunked by the community. There was a lot of technical flaws in that analysis, and there was basically live peer review within essentially 24 hours. The authors actually end up retracting the preprint. Now, my guess, if they went directly to a journal without going through the preprint route, my guess is that would have been published in journal. They would have found a journal that would have published that work. I am certain about that. And it would have gone through peer review. We know that peer review is very poor at actually filtering bad science. It's good at improving science. It's good at that. But it's not very good as a filter, because essentially you can go and keep submitting to one journal after another. And there's now so many journals that are, let's not say predatory, but semi-predatory, where the incentive to publish is so high that they just really place the bar very low. So I think the damage that would have been caused by that paper coming out in a peer-reviewed journal would have been much, much worse than what happened when it was posted as a preprint with the red flag. Every preprint is flagged as saying this is not peer-reviewed, warning. And now you see more and more in the news comments that this is a preprint, this hasn't been peer-reviewed. People even mention that in newspapers articles these days. So my answer, Brenda, is simply that fake science is getting published everywhere, and peer journal reviews are full of fake bad science. And so I don't see this as necessarily an issue with preprints. I actually find it that with preprints, we're not claiming it's correct. There is a flag actually that this hasn't been peer approved. So in a way, the damage should be less than with journals.
Then thank you for that. I see a question from Zander Mayberg about publishing posters, whether you need permission from conference organizers. And a second question.
I hope not. Yes, Zander, yeah, thank you. But I hope not. It's your poster, so I think unless the organizer, and I hope organizers of conferences will not move in banning posting of posters, but it's really your poster, and I think I don't really see why they should prevent you from doing that. This is, I'm sure all of you use Sci-Hub. I'm not advising you to use it. It's against the law, but we all know about civil disobedience. So I think the publishing world right now is so messed up. We are all working for free for some publishers to make money, and sadly this applies to conferences. Obviously not your type of conference, which is organized by a scientific society, but there are more commercial conferences. So I would actually advise you to fight really hard if you're somehow involved in a conference that would prevent you from publishing your poster. I think you should exert your own judgment there and not be constrained by the rules.
Then I see a question about whether one can revise preprints after peer review. Yeah.
Yes, absolutely. Yeah. In fact, let me even expand on that. We recently started doing even something else, where we actually post a preliminary version of the paper as a preprint. Let's call it V1. And we know that we're still doing additional work before submitting to a journal. So actually the version that we end up submitting to the journal is not the first one, but maybe the second or even third version. And so this is a way of actually posting a paper that is not 100% complete, let's say 80% complete, getting feedback from the community, having time to do more experiments, and then actually posting a version 2 that we then submit to a journal. We're in the middle right now of actually doing that, where we have a paper that's been out there as a preprint since, I can't remember now, maybe May or June, and we're now posting the second version that we will submit to a journal. So yes, absolutely, you can post multiple versions. However, you cannot remove the old ones. So this is the whole point with the DOI, is once you post it, it's there for posterity.
I see a question from Renee Hein, who asks, instead of putting responsibility on the shoulders of individuals, would it be possible to involve societies to help purging the peer review system and use a guided open approach?
Yeah, of course. I mean, I think everyone should have responsibility. My motto in life is that, you know, evil prevails when good people do nothing. So we should all do our bit, obviously, including societies. And I think your community is very progressive, and I know what Bernard does and FABI does, for example. And I think this is all very good. We should all have this, contribute our bit and not expect to others to lead. If we all did a little bit, then the world would be different. So yeah, of course, I mean, scientific society should also help with these issues. But and personally, I actually think the funding agencies have the most responsibility. I don't understand why funding agencies do not mandate preprints. I really don't understand that. I mean, they go through now this completely ridiculous, complicated Plan S, process, which is actually basically just putting more money into the pocket of the publishers. It's very complicated. It's just doing exactly the opposite of what should be happening. And they could solve all these problems by mandating preprints. Another thing with preprints, by the way, this is a good one. I like this because this is kind of marking a point against the publishers. When you post your preprint, make sure you publish it as CC BY, which means that you're basically giving away the copyright for the figures and all that, and anyone can reproduce them as long as it's not for commercial uses. But what that means that even if later on the paper is published in a journal, anyone can still use your figures because they're already published as a CC/BY, as a preprint. So think about teaching and all that. I'm actually involved now in some lectures with our university, and then they're going through the trouble of telling me, oh yeah, well, this figure you took from this paper, so you have to ask for copyrights, and they ask for $300, and it's just ridiculous. So if we all did that, if we all posted our papers as preprints as the CBY, then we wouldn't have this problem. Anyone in the world can actually reproduce and use that material for teaching and all that. So bottom line is, I don't understand why funding agencies, I don't know, I don't know what's going on there. It's just too conservative. They should mandate preprints, period. And then, after that, people can do whatever they want, publish it in wherever, doesn't matter. But if it's a preprint, anyone can read it. It's free also. BioRxiv is supported through foundations now. Thank you, the Zuckerberger Foundation. They're funding them for long term, actually. So there's no issue there with money.
I think thank you for helping us with more perspective on that. And I also believe we're going to see more and more in the coming years. And it really, as you pointed out, addresses many of the concerns many of us have in plant pathology and plant pandemics and how information is shared there. We have run out of time. There's one last question that I'll highlight there from Brenda, because it's not only about sharing the data, of course, and the papers, but also the biological material, as you just pointed out in our wheat class. It's really important. And the issue with the Nagoya protocol that makes it very difficult for us to share. As she points out, even in South Africa, to get permits to share material internationally is tricky. Can you comment on that for us, please?
Gosh, I mean, Brenda, what can I say? I mean, this is really a complicated issue, and I share your pain. I mean, we're all struggling with Nagoya, and it's a big can of worms. I don't really have a simple answer. I mean, again, I think the part of the issue right now is that sometimes these complications are being used as excuses for not sharing or for keeping data secret in a way, which I think is getting harder and harder to do. But I think Nagoya is often used as an alibi. And in our experience, I mean, in projects we've been involved in with Bangladesh, with Zambia now, this hasn't been necessarily an issue. So when there is a willingness to collaborate with local scientists, I think generally there is a solution that's being found. But I see your point. This is Nagoya has been complicated, let's put it this way, as you very well know, I think.
Clearly many things, and as you say, for bad things to happen requires good people to do nothing, and a strong call that you make today. So Glenn, thank you again for a wonderful talk and for that strong call. But I think, in terms of just coming out of an International Year of Plant Health, it's exactly what I think we should be doing in an SASPP like this today. So thank you very much for that. Let's do a round of applause in the room here.
Thank you very much, Bernard. I just want to add that I will answer the questions in the chat, and also that it's very very motivating for me to interact with your community and get the sort of feedback I'm getting. Thank you so much.
And we hope we can redo this meeting in person as soon as at the beginning of next year, we hope. And we hope that you will be with us then. So thank you very much, Sophia. We're gonna stop now for 10 minutes, I think. There's a little bit less than 10 minutes left. And we will reconvene at 1:15. Reconvene at 1:15 so we get back on the program. I look forward to seeing all of you back. Just get a leg stretch a bit.
Good morning, everyone. My name is Amun Hamabaha, and I will be chairing the scientific part of this conference. Welcome, and I hope that you enjoy the program. The first speaker that I would like to introduce is Megan Dutoy from Stellenbosch University. She recently changed her name, so I guess that she got married at some point recently. And she is working with Altus for You on Panama disease, and she will be sharing with you some of the things that she's learned about the diagnostic of this disease. Joanne, will you share the presentation?
Good morning. Today I'll be presenting on the quantitative detection of economically important FOC strains in Africa. I will begin with a brief introduction. Firstly, about the importance of bananas in Africa. Along the vast continent of Africa, there are different varieties, including East African highland bananas and plantain, which provide important local staples to many people, as well as local dessert bananas and Cavendish, which are used as a source of income, as well as in the production of beer and other snacks. The banana industry in Africa and globally is under threat by a devastating disease called Fusarium wilt. The causal agent is known as Fusarium oxysporum forma specialis cubens, or FOC. FOC has characteristic spores, including macroconidia, chlamydospores, and microconidia, shown here. FOC can be classified into three races, race 1, race 2, and race 4, depending on the host cultivars that they affect. In Africa, race 1 and 2 isolates can be put into the same lineage, known as lineage 6, based on genetic traits. Race 1 and race 2 are able to infect and cause disease in cultivars, such as Gros Michel and Blue Go, respectively. FOC race 4 is the most pathogenic and can cause disease to the Cavendish variety, the most important export banana of our time, as well as many other cultivars susceptible to race 1 and 2. Race 4 can be split into two variants, a tropical variant, which causes disease in both tropical and subtropical conditions, and a subtropical race 4, which can only cause disease when the host plant is under stress, such as during the winter in the subtropical regions of the world. The pathogen FOC is spread in different environmental sample types, including infected plant material, infected water, and soil. Its highly resistant chlamydospores are particularly difficult to manage, as they can be viable for decades. Preventing the spread of FOC is vital, as once established, the pathogen is very challenging to manage. If the pathogen does enter a banana plantation, it is possible to keep production above an economic threshold by using tolerant varieties. However, these varieties need to be supported by methods such as biocontrol or crop management that will keep the inoculum below a certain level. The management of FOC is much more successful if the pathogen is detected early. For this reason, early detection is one of the primary focuses in the management of Fusarium wilts. A common method for detection of pathogens in environmental material is quantitative PCR. It is very sensitive and accurate and works in much the same manner as normal PCR, with different cycles of temperature and phases such as denaturing, primer annealing, and replication. Rather than visualizing the end products via gel electrophoresis, fluorescence is used to track the amplification of target DNA in real time. Markers are available for the detection of FOC TR4 with qPCR. However, there are not markers for subtropical race 4 and lineage 6. This brings us to the methods and materials. To design primers for our target FOC isolates, we used a database containing the sequences of hundreds of FOC isolates as well as closely related specimens. We then tested the specificity of all the primers, and then if primers were suitable and specific in a qPCR setting, we made standard curves which enable absolute quantification from an environmental sample. We then did validate our assays using the MyQ guidelines, and lastly, when the assays were validated, we could use them to test environmental samples that were naturally infected with FOC. Looking more closely at the first step, once primers were designed for FOC tropical race 4, STR4, and lineage 6, which are all of economic importance in Africa, we tested them using PCR. This was done using 35 isolates which represented the diversity of FOC. If the primers were suitable for PCR and proved specific, we would then test them in a qPCR setting. This was done with 127 isolates, including many FOC isolates, as well as many non-pathogens and closely related Fusarium oxysporum and Fusarium isolates. To make a standard curve, we take target DNA, for example, if we were making a standard curve for tropical race 4, we would isolate 20 nanograms per microliter of target DNA from the mycelia of our target pathogen, and then dilute it in a background of DNA, which was isolated from the environmental background, so either water, plant, or soil material that is void of the target pathogen. In our case, we used a 4 times dilution series with different concentrations. We then used our dilution series in a qPCR run and told the software what concentration of DNA should be at each point, depending on the dilution. The qPCR software then correlates this concentration that we gave it to a CT value, or cycle threshold. This is the cycle of DNA amplification at which time a sample's fluorescence exceeds that of a background level. After the CT and concentration of DNA are correlated, the software gives a rating for the standard curve in terms of efficiency and R-squared value. The efficiency should be 1 if there is an exact doubling of DNA after each cycle, and the R-squared value should be near 1 as it is a correlation index, letting you know how well the points that you have plotted fit the line. Moving on to the validation, the reproducibility and repeatability of each qPCR was evaluated. That is the variation in terms of standard deviation between the same DNA sample run on 2 different days, as well as the same DNA sample run in triplicate. Then we looked at the limit of detection and the limit of quantification. The limit of detection is the lowest DNA concentration at which detection is certain for a particular qPCR assay. The limit of quantification is the lowest DNA concentration at which quantification is accurate, or at which the standard deviation is below a threshold. Lastly, after the first 2 validations, we tested the specificity of our assays when FOC infected plant, water, and soil samples were used. This was done to ensure that specificity was not negatively affected by the environmental background. Coming to results, the gene regions of the best performing primers for lineage 6 was a 98 base pair region within RPC2. For TR4 was 157 base pair within a supercontig, so a gene region within our database at Stellenbosch University, and a 235 base pair within a hypothetical protein was also used for SDR4. The first two markers proved specific throughout testing, both in PCR and qPCR settings. However, the FOC SDR4 markers, although specific in PCR, did pick up on non-target isolates in qPCR. These included a isolate of Fusarium proliferatum and Fusarium saccharide, and two non-pathogenic Fusarium oxysporum isolates. We therefore decided to use the SDR4 marker in a multiplex with markers that target FOC69 gene. This is a gene that is associated with pathogenicity in FOC. Coming to the standard curves for the lineage 6 TR4 and the SDR4 multiplex, the efficiencies in R-squared values ranged from 98 to 103% and 0.99 to 0.999 respectively. These met our requirements and were suitable for absolute quantification. Looking at the repeatability and reproducibility, it was shown that standard deviations were below 0.35, and so the assays were acceptable according to this standard as well. The limit of quantification and limit of detection were between 10 to the minus 2 and 10 to the minus 4, and 10 to the minus 3 and 10 to the minus 5 respectively. There was a higher limit of quantification and detection in the water matrix compared to the other two matrices. The lineage 6 and tropical race 4 qPCR assays remained specific when we tested them against DNA from FOC-infected plant, water, and soil samples. This brings us to the discussion. The qPCR assays for lineage 6 and TR4 are useful and could be used for the rapid detection of FOC inoculum in quarantine units or testing facilities, which can provide data on Fusarium wilt dispersal and perhaps improve containment strategies in Africa and on a global level. Resistance screening of banana cultivars could also be an avenue where this assays could be used, as well as investigating soil inoculum levels, which are necessary for disease development, specifically when tolerant varieties are used. The qPCR assays for subtropical race 4 are useful for identification and for quantification in the laboratory, but are, however, unsuitable for absolute quantification in environmental samples, as the primers for the 6-9 region will also detect pathogenic FOC isolates that are not SDR4. And from research, we can see that subtropical race 4 is often also present in soil and plant environments. For the detection of any of the FOC isolates in soil and water environments, sampling still needs to be optimized, as the pathogen is often present in low concentrations and is heterogeneously distributed in these environments. Thank you for listening. Please feel free to ask any questions, as the time was limited, and so the explanation is quite concise. Thank you.
Thank you, Megan. So if you have any questions for Megan, you are welcome to either raise your hand and speak up, or type something into the text box. So I can see here Brenda has got a question about from which chromosomes did your primers come from? Are these core or accessory chromosomes? Megan, are you here?
Okay, I couldn't unmute myself. So when we looked at, for primer design, we looked at a lot of different regions, but because the database was organized and not all of the regions are very well described, what happens is you sometimes can't be sure which chromosome it's supposed to be located on. So for some of them, it was easier to describe, but for many of them, so for example, the TR4 one, all I could find was the region within our database that it was located in. and we're not sure of the exact function of that region. So we would have to go much further to actually figure that part out. But I think it's a good question, and there is a lot more work that needs to be done in figuring out which gene regions are responsible for which function.
So Neriman commented that the taxonomy of this whole Fusarium is very, very messy. And can you comment on your races, how they are actually linked and related in the Fusarium oxysporum species complex?
So I would agree, it is a bit messy. And in my honors project, I actually also worked on Fusarium oxysporum isolates and taking them from soil around South Africa and trying to organize them in a phylogenetic tree. And what happens is even the former specialis aren't always grouped as you think they should be. So it is very challenging to have an exact answer once again. But I think working on our specific pathogen, if you can prove that it's pathogenic to your host or your crop, that's more what we look at in terms of our isolates and how to separate them from Fusarium oxysporum, other than genetic differences, which we are continually working on. So we're always trying to get more markers that can separate it from the vast majority of Fusarium oxysporum members. But once again, the problem with doing this is you have to then test your markers against so many different isolates, and not everyone has all of them. So we have a large collection, which we're fortunate to have, but that doesn't mean that there isn't another Fusarium oxysporum isolate that could pick up when it shouldn't. So I think with such a difficult pathogen and so many different isolates, you have to be very careful when testing for specificity and when describing it. You can never say for sure that nothing, no non-target is going to pick up with your markers.
Yeah, that's a good answer. So any other questions for Megan? Let's see. In the chat, yeah, well, Vishan is commenting actually that there are only 6 genes on the accessories genome.
No, the 6 genes, the SIS.
Yes.
Ah, I see. Yeah.
So with the 6 gene, that one we didn't use by itself. We used it in a multiplex with the SDR4 isolates. And I do think with that assay, I'm not as confident as the other 2 for that reason. And it also makes it tricky to use because now you have 2 markers, which should make you more confident that it's specific, but it also means there's more of a chance that you will sometimes have difficulty interpreting results, and you actually have to test it a lot more thoroughly, which I think we should do given the 6 genes. Yes.
Good. Okay. Thank you, Megan, for a nice presentation. Our next speaker is Kira Lynn, and she's from the University of Pretoria doing her PhD in genetics, and she's done all her other degrees in genetics as well. Kira is fortunate in the sense that she can travel the world with her research funding, which comes from an Indonesian company. And in this specific case, what she's going to be talking about is actually a beetle from Indonesia, which she's been studying, and also the associated fungi of this beetle. So over to Kira Lynn.
Good morning, ladies and gentlemen, and thank you so much for the opportunity to present some of my work focusing on investigating ambrosia beetles that were infesting acacia carpa plantations in Indonesia, as well as some of their fungal mutualists. So ambrosia beetles are wood-boring insects. They come in a variety of shapes and sizes, as depicted by this picture. And they spend almost their entire life cycle within the xylem of suitable plant hosts. They're able to survive in this nutritionally poor ecological niche due to their obligate nutritional symbiosis with fungi. So as depicted in this picture, here are some ambrosia beetles. And this is just a normal MEA plate. And we put these ambrosia beetles within this plate, and they began tunneling and cultivating their nutritional fungal associates and survived. You can even take a closer look into a tunnel and you can see here that the fungal mutualists are growing. And we were even able to capture a beetle in the process of consuming its fungal associate. So ambrosia beetles transport these microbial mutualists within specialized organs termed michangia. And these are depicted by the yellow outlines and can be located on different parts of the beetle body depending on the ambrosia beetle species. So ambrosia beetles can be associated with a wide variety of fungal species. However, the fungal mutualists found within their michangia are distinct and defined as ambrosia fungi. In the ecosystems of their tropical and subtropical native ranges, most ambrosia beetle species live in decaying wood and generally do not have any economic consequences. However, changes in their ecological interaction, particularly their accidental introduction into non-native regions, have resulted in a few ambrosia beetle species, particularly those within the family Xylobarini, to become some of the most important pests on various economically significant plant hosts around the world. The shift in virulence of microbial mutualists of ambrosia beetles from benign nutritional mutualists to phytopathogens isn't really well understood. What is clear is that an increase in the number of these ambrosia beetle-related diseases is usually associated with activities that facilitate new encounters between the beetle-fungal symbiosis and novel plant hosts. More specifically, the emergence of these diseases is largely attributed to the interaction of the beetle's microbial mutualists and novel plant hosts that lack co-evolved adaptation. Therefore, identifying the fungal associates of these ambrosia beetle pests and determining their diversity and pathogenic potential is pivotal to understanding their disease biology. Most of the research conducted on several of the most prolific ambrosia beetle-related diseases has been conducted in their non-native environments after they've been accidentally introduced and have caused significant economic damage, as is demonstrated by several of these publications below. Thus, the level of diversity of these invasive beetle species and their microbial mutualists is most likely underrepresented. This hypothesis was reiterated by a recent study investigating members of the Ulacea fornicata species complex in their native range of Taiwan. In this study, it showed that the relationship between the species in the Ulacea fornicata species complex and the ambrosia fungi they cultivate is likely more diverse and promiscuous than previously described in invaded areas. Thus, understanding the ecology of ambrosia beetle pests in their native environment can provide a basis for better management strategies that can be implemented to mitigate their impact if they're accidentally introduced into new non-native regions. So this brings us to our disease problem, in which a gradual increase in the number of ambrosia beetle infestations in non-native Acacia crassicarpa plantations, which are grown for pulp and paper, was observed in their presumed native range of Indonesia. Not only is this emerging disease outbreak a cause for concern that needs to be further investigated, but we don't know why there's been this sudden gradual increase. We don't know if it's a native beetle or an introduced beetle. But this disease paradigm also provides a really great opportunity to understand the ecology of ambrosia beetle pests in their native environment and expand our knowledge regarding the diversity of the fungal species they are associated with. So the aims of our study are therefore to identify the ambrosia beetle species that are infesting these Acacia crassicarpa plantations, as well as identify their fungal mutualists, with a particular focus on fungal mutualists that have been previously reported as plant pathogens. So to do this, we were fortunate enough to head down to Indonesia, where we surveyed 5 geographically separated compartments of 2 to 3-year-old Acacia crassicarpa trees. After identifying heavily infested trees, we felled them, and we took these felled trees back to the laboratory. Here we extracted the beetle brood galleries, and from these beetle brood galleries we also extracted ambrosia beetles. From both the brood gallery as well as the ambrosia beetle michangia, we did fungal isolations. We then performed morphological identification of the ambrosia beetles, as well as the fungal isolates we collected, and then also did phylogenetic analysis to identify them. There's approximately 3,400 known species of ambrosia beetles that reside in 11 tribes. But based on our results, we'll be focusing on the Uelacea fornicata sensillata complex that consists of 4 sister species that have recently been taxonomically redefined, as well as some closely related species to this complex. So based on our results, we identified Uelacea brevis as well as Uelacea similis as the most abundant beetle species infesting these Acacia crassicarpa plantations. They were quite easy to distinguish morphologically, with the most prominent feature being the spines on the back of the Uelacea similis to distinguish it. They interestingly also differed ecologically, with Uelacea brevis acting more as a primary borer, meaning it would often infest seemingly healthy trees, whereas Uelacea similis appeared to act more in line with typical ambrosia beetle behavior and was a secondary borer that could only attack hosts that were previously colonized or weakened in some way. Phylogenetically, these 2 ambrosia beetle species also showed quite a high amount of diversity, with Uelacea brevis in green and Uelacea similis in blue. We identified 3 novel haplotypes of Uelacea brevis and 4 novel haplotypes of Uelacea similis, with both these beetles and their haplotypes being widely distributed across the sampled Acacia crassicarpa compartments. Moving on to the fungal mutualists, we were able to identify 3 novel Fusarium associates of Uelacea brevis, the most abundant of which was Fusarium ruricinem, as indicated by the thick red arrow. The abundances of these Fusarium associates differed and is indicated by the thickness of the arrows. We were able to identify 5 novel Fusarium associates of Uelacea similis. Again, these associates varied in their prevalences, with Fusarium 1 being the most abundant Fusarium associate isolated from Uelacea similis. As Uelacea similis appears to be a secondary borer, we believe it has the ability to acquire Fusarium associates of other closely related beetle species, and is why it has a more promiscuous relationship with its fungal mutualists than Uelacea brevis. We were also able to identify another Fusarium associate, but we could not confirm its beetle vector, as it was isolated from the brood gallery that was inhabited by both Uelacea brevis and Uelacea similis. In conclusion, we were able to identify 7 novel Fusarium associates of these 2 Uelacea beetle species, of which 2 of these Fusarium associates were shared by both Uelacea brevis and Uelacea similis. So this study not only gave insights into the diversity of 2 Uelacea beetle species infesting Acacia crassicarpa plantations in Indonesia, but also gave insights into the diversity of Fusarium associates that they cultivate. As we previously stated, this is quite important, as the key factor in disease outbreak seems to be the interaction of these beetle fungal mutualists with novel plant hosts that lack co-evolved adaptation. It also emphasized that this beetle fungal symbiosis appears to be more promiscuous in nature than previously described in invaded areas. And because of this, these beetles seem to have the potential to swap symbionts of closely related beetle species. And this could result in new disease outbreaks, as different Fusarium associates could have different pathogenic potentials. Thus, investigating these Fusarium farming Uelacea beetle species that have emerged as pests in their native environments can really be useful and give insight into the pathogenic potential these beetles can have and inform future quarantine strategies. And a really good example of this is what's happening in South Africa. So in South Africa, a sister species of Uelacea brevis, which was Uelacea fornicatus, otherwise known as the polyphagous shothole borer, was introduced to South Africa from Southeast Asia. So by investigating these disease outbreaks in their native environments, we can hopefully mitigate the risk of new introductions of not only new beetle species, but also novel haplotypes of beetle species that are already present in non-native environments. as well as new fungal mutualists that these beetle species or novel haplotypes can harbor. And as we said earlier, these new introductions can result in the swapping of fungal mutualists, creating greater disease outbreaks. But also it's been shown that these Fusarium mutualists have the ability to recombine, which could wreak havoc on resistant breeding programs. In conclusion, I would also like to emphasize the importance of taxonomy that we discovered in the study. So during the period of this investigation, the identity of the beetles I was investigating changed twice. And this really is important to try and communicate scientific knowledge in the sense of informing quarantine strategies, of knowing what beetle species are already present in non-native environments and what beetle species that you should be on the lookout for. And with that, I would really like to say thank you. Thank you so much for listening. Thank you for the opportunity. Thank you to my funding bodies, to my supervisors, to my Indonesian collaborators, and to everyone that just helped me along the way. Have an awesome day. Thank you.
Thank you, Kira. Kira, are you here?
Yes, I am. Can you hear me?
Yes, loud and clear. So Grisel is asking, do you know of any studies which determine the identity of fungal associates by means of a culture-independent approach, and do they identify a greater diversity?
So I do believe there has been some studies. I don't know them in depth, but I know they're used by means of non-culturable, where they take isolations and they, I don't know the exact methodology, but they identify OTUs. And then based on those OTUs, they identify that diversity of fungal associates associated with those beetles. So there are studies that do that. And they also focused on what was associated with particularly the mycangia or with the beetle as a whole. So there are, I'll have to go look at some of my papers, but there definitely are research focusing on this.
Okay, that's a good answer. Bernard has a question, not an answer.
Thank you, Ahmed. Kira, thank you for a good talk. I'm wondering about the swapping of the symbionts. Do we have an idea of how frequent that happens? And perhaps I can look at the question from two directions. What stimulates such a swap, or what maybe hinders it from happening more often?
That's a really interesting question, and one that I have pondered. So I don't think there is a particular way of measuring the rate or how often swaps occur. And I think that's particularly, as I said, because most of this research was conducted at present in areas where these beetles have become invasive. So there's been this kind of bottleneck effect. So I don't think you really can investigate that. But with more and more research, what they're realizing is that different related Eualaceae species can harbor similar symbionts. So again, with the rate, I don't know. With the fact of what inhibits it or encourages it, a lot of these mycangia structures have selective pressures. So they can favor associates depending on the secretions and the environment that these beetles are in. So it also has been hypothesized that some of these associates are better adapted to infesting certain plant hosts. So therefore those beetles could acquire those hosts. It could simply be temperature based. So some studies have shown that some of these beetles in different seasons, their fungal associates or mutualists abundances vary depending on the season. So I think it's a really interesting question, one that I haven't investigated in depth myself, but I'd really be interesting to kind of look at.
Thank you.
Okay, thank you, Kira. So our next speaker is Patience Chakagupta, and she is from Wits University. She's getting a research fellowship there. And she's also a mom and a biotechnologist and a biochemist. And she will be talking to us about virology.
Good morning to you all. I'll be presenting on the topic protoplasts in CRISPR, hand in hand towards high throughput gene screening in cassava. This was carried out as part of my postdoctoral fellowship in the plant biotech lab at FIX. Now, one of the projects in the plant biotech lab is the study of the interaction between cassava and cassava mosaic viruses. And in particular, we're looking at South African cassava mosaic virus. This is a bipartite Gemini virus. It contains, its genome is divided into two, DNA A and DNA B. And when it infects cassava, it produces symptoms of mottling, mosaic, as well as wilting. And in extreme cases, it can cause up to 100% yield loss. Various cassava genes have been implicated in the response to cassava mosaic viruses, and this has been explored in literature. And a lot of these studies, they used a reverse genetics approach of gene screening during infection. And in our lab, we were using a cumbersome protocol for reverse genetics gene screening, which involves the production of somatic embryos, the proliferation of riboembryogenic callus, the transformation of this callus, and the selection of transformed plantlet. This protocol takes at least 20 weeks if everything goes according to plan, and it is extremely expensive due to specialized media and assay chemicals, as well as dedicated growth rooms and chambers. So we're looking for a faster and simpler approach to gene screening, and we decided to use CRISPR, which is a fast and simple gene editing tool with proven efficacy in reverse genetic studies, in protoplasts, in virus infection, and for high throughput screening. And we decided to combine CRISPR with protoplasts, which are viable cells that lack a cell wall. They can be simply isolated and transiently transformed. They are reliable for implanter activity correlation, and they have proven efficacy for gene characterization. So we thought this might be a workable idea, and we decided to try this with reference to an implicated ubiquitin E3 ligase gene known as MEE3L. And the reason why we targeted the ubiquitin system is because the ubiquitin system is known to be reprogrammed and redirected by Gemini viruses such as cassava mosaic viruses. So we were working with 3 different cassava genotypes, CV6444, which is the model cultivar, T200, the susceptible landrace, and TME3, the tolerant landrace. And the idea was to co-infect or co-transform protoplasts with SACMV infectious clones and with the CRISPR silencing vector targeting MEE3L. So we managed to isolate our protoplasts using an enzyme-mediated protocol, and we carried out a viability assay using Evans blue dye staining. We also quantified our protoplasts and assessed their quality using flow cytometry. And we carried out a transformation assay to check whether the eGFP encoded in our CRISPR silencing vector is being expressed. So when we sequenced the genomic sequence of MEE3L and aligned it with the reference genome AM560-2, we found that the susceptible MEE3L homologue contains a 53 base pair insertion mutation. And this insertion mutation actually codes for a stop mutation at amino acid residue 141. And this stop mutation is just upstream of the ring finger domain, which is where the ring E3 ligase activity resides. When we carried out computational prediction of the structures of our MEE3L homologues, we found that the T200 homologue is structurally different from the other two, and it lacks a zinc binding motif, unlike the other two. And we also found that the T200 ME3L homologue is evolutionarily distinct from the other ME3L homologues. When we assessed relative SACMV DNA accumulation in cassava protoplasts, we found that the accumulation is much greater in the susceptible landrace, and it is even more pronounced when the ME3L gene has been silenced. We then also assessed the relative expression of ME3L, and we found that it is significantly upregulated in tolerant cassava protoplasts, and even more so when it has been silenced using the CRISPR silencing vector. We then looked at the genomic sequences of our ME3L homologues in the SACMV infected protoplasts, and we found that there were mutations in these homologues that translated to stop mutations in both the susceptible and the tolerant, just upstream of the ring finger domain. And we also found that in the tolerant landrace, there were multiple mutations in the SACMV infected variant. So we have found that African cassava genotypes are amenable to enzyme-mediated protoplast isolation, PEG-mediated transformation, and high-efficiency CRISPR-mediated gene editing. We have also found that the susceptible cassava genotype encodes an inactive ME3L where the ring finger domain is silenced. So the question is, does this mutation contribute to susceptibility? And is this genotype still adapting to SACMV as it co-evolves with the virus? We also report that the SACMV DNA accumulation in the 3 protoplast genotypes correlates well with previously reported in-planter results. The response of ME3L to SACMV infection and gene editing in the tolerant genotype suggests its involvement in the response to SACMV. Because we see that SACMV induces multiple silencing mutations in this tolerant ME3L. So our findings indicate that ME3L is responsive to SACMV infection and may play a role in susceptibility and tolerance outcomes. Personally, the greatest outcome for me was that we managed to reduce the timeline for gene screening from plus 20 weeks to just 8 weeks, which is really great. There are some limitations to this protocol. Protoplasts are independent cells and they have short-lived viability. So this protocol cannot be used to study cell wall-related genes or cell-to-cell signaling or intercellular movement. It also cannot be used to study long-term responses or long-term stability of CRISPR-induced gene edits. We isolated our protoplasts from leaf mesophyll, so we feel that leaf mesophyll protoplasts may not be suitable to correlate responses in other organs such as roots or flowers. It is important to validate the results of this protocol in planter by overexpression and virus-induced gene silencing of targeted genes, as well as functional and interaction studies to confirm the specific roles that these candidate genes play in the host virus interaction. So we published this work in the Virology Journal with the title, A Cassava Protoplast System for Screening Genes Associated with the Response to South African Cassava Mosaic Virus. We are expecting to target more candidate genes for association with the response to SACMV. And so far, we have targeted quite a number of RNA interference genes, such as your organofamily, your RDRs, as well as your SGS3s. And we have seen that these are significantly downregulated during SACMV infection. And particularly the AGO4 also contains numerous SACMV-induced mutations as well. We are also exploring generation of CRISPR-transformed plants for implanted validation. And that brings me to the end of my presentation. I'd like to thank Prof. Chrissy Ray for hosting me at FITS and Prof. Fundus Curran for providing us with the CRISPR silencing vector. And I'd also like to acknowledge the support of my lab mates in the Plant Biotech Lab, as well as funding from the National Research Foundation. Thank you all for your time.
Thank you, Patience. Are you here?
Yes, I am here. Can you hear me?
I can hear you. I want to ask a quick question. How do you then identify and select genes that that you're going to screen with your system, which is a great system, by the way?
Sorry, can you repeat the question? I didn't hear the first part.
How do you find how do you select, how do you choose which genes you're going to use for the screening?
So I already mentioned in the presentation that several studies have been done on genes that are on identifying candidate genes that are involved in the response to cassava mosaic viruses. Some studies have specifically looked at the disease resistance loci in cassava. Some studies actually took a transcriptomic approach. So we just sort of retrieved the genes that we think would be interesting from all those studies. And particularly, we're looking at resistance genes, the ubiquitin system genes, as well as the genes from the host RNA interference system, because these are known to interact very much with Gemini viruses upon infection.
Okay, and we also have a question from Dave, and he asks, do you have an idea on how this virus of yours induces targeted mutations in cassava genes?
No, so we have no idea how it does this. This was an interesting finding from our research, and it obviously needs more study. But we do know that that the Gemini virus can actually enter the nucleus, and that some of the double-stranded breaks are induced by Gemini viruses. So this is known, but we don't know the actual mechanism by which it occurs.
All right, thank you, Patience, for really wonderful.
Thank you very much for listening.
Okay, so our next speaker is Minette Harpenbach. Minette is shared by the University of Stellenbosch and the University of Pretoria. She works at both. And the outstanding fact about Minette is that she won the Melbourne Hall Stakes last year. And unfortunately, due to the alcohol ban, we can't find a challenger to this, but next year you have a chance to win it from her, the trophy. So Minette is working on Terranspheria destructans, which is a eucalyptus pest, and she will tell us a little bit more about the population genetics of this fungus.
Thank you for giving me the opportunity to present my research today. I will be presenting on the population biology of a globally important eucalyptus foliar pathogen called Terranspheria destructans. Then I will also discuss a new tool that we developed for rapid identification of this fungus. Terranspheria destructans causes severe shoot and leaf blight, which leads to the defoliation and dieback of newly planted eucalyptus trees, especially those that's planted outside of their native environment. Under moist conditions, asexual fruiting structures form below the stomata, and they exude asexual cornelia that gets dispersed to the neighboring trees. Terranspheria destructans was first discovered and described from Indonesia in 1995, after which it rapidly spread to other tropical and subtropical eucalyptus growing regions. It was reported to cause disease in Thailand, Vietnam and East Timor, China, and Laos. And then in 2015, Terranspheria destructans was discovered causing serious damages to eucalyptus plantations in Zululand, South Africa. This was the first case of this pathogen occurring outside of Southeast Asia, and it has since spread to all eucalyptus grandis regions in South Africa. Shortly thereafter, The pathogen was reported to cause disease in Malaysia. So as you can see, this pathogen is currently limited to Southeast Asia as well as South Africa. And even though this pathogen was discovered in 1995, little is known regarding its genetic diversity, its origin, or its dispersal pathway. Therefore, we investigated the genetic diversity, recombination potential, and global relatedness of 7 populations with a specific focus on our South African population. And because this pathogen has spread so rapidly in South Africa, we also assessed the developed microsatellites from the study as a rapid and cost-effective diagnostic tool. So we sequenced 3 genomes and we used them to develop polymorphic microsatellite markers. We used 10 microsatellites and applied them to a global collection of isolates. The collection consisted of isolates from 7 populations, of which 6 were from Southeast Asia and 1 from South Africa. The South African population consisted of isolates from multiple sampling sites in KwaZulu-Natal and 1 sampling site in Mpumalanga. So we found 38 genotypes, of which 37 were limited to their specific population. Only 1 genotype was shared between some of our populations, specifically isolates from Thailand, all of our isolates from Vietnam and South Africa, and 2 isolates from North Sumatra. These 2 isolates were coincidentally the 2 oldest isolates in our collection, and they were sampled in 1995 from the original disease report, making this not only the only shared genotype, but also the oldest genotype in our collection. So if individuals are regularly moved between populations, we would expect more genotypes to be shared among our populations, which wasn't the case here. The Malaysian population had 20 genotypes, which was far more than any of the other populations. This is very surprising because new populations would not be genetically diverse. It can even be clonal, as we see in South Africa, because they have undergone a bottleneck and have not had time to evolve. On the other hand, we can see that our 2 oldest populations in Indonesia had the second most genotypes, and this would be expected from older populations. Here we show the genetic distance among the genotypes in a minimum spanning network. Each node in the network represents a genotype. The genotypes generally separated according to their sampling region, indicating independent evolution after the pathogen was introduced into their respective areas. And the Malaysian population looked a little bit different. We see that these genotypes did not intersperse with any of those from other locations, and they were generally separated from the populations by a larger genetic distance. To assess the population structure, we used 2 methods, and both assigned individuals into the same 4 clusters. Again, we can see that our isolates grouped according to their geographical location, with the 2 Indonesian populations in cluster 1, populations with the shared genotype in cluster 2, isolates from Malaysia in cluster 3, and our Chinese isolates in cluster 4. The Malaysian cluster had high levels of admixture, particularly with the Chinese and the South African, Thailand, and Vietnam clusters. And the Malaysian population also had the highest genotypic diversity of 20%. This was nearly double the recorded diversity of our older populations found in Indonesia. All other populations had low genetic diversities, which was not surprising since 89% of isolates belonging to these populations were clonal. Our findings of low genetic diversity and high proportions of clone is supported by the reproductive strategy of destructants that we investigated in an earlier study. The pathogen has an outcrossing mating strategy, which means that 2 individuals of opposite mating types are needed for sexual reproduction to occur. Using primers that targets the conserved region of mating type genes, we could investigate the mating type distribution in all of the populations. All populations, except for Malaysia, significantly deviated from the expected 1-to-1 ratio, confirming our previous findings that sexual reproduction in these populations are very unlikely. And in Malaysia, the ratio did not significantly deviate from the expected 1-to-1 ratio. Together with the multiple genotypes, this indicates that sexual reproduction could be occurring in Malaysia. If we look at South Africa, the South African population consisted of a single genotype, and now we also see a single mating type, which just further confirms that South Africa is completely clonal. So if we look at our 2 different reproductive strategies, all investigated populations except for Malaysia had low genetic diversities, skewed ratios of mating types, no evidence of recombination, and a large number of clones, indicating that the asexual state is the dominant mode of reproduction and dispersal of this pathogen in these areas. For Malaysia, we also see a high number of clones, indicating that asexual reproduction is present in this population. This is consistent with the fact that only the asexual state is known for this pathogen and is also regularly found in heavily diseased trees. For sexual reproduction, the Malaysian population had the highest genotypic diversity, even distribution of mating types, evidence of recombination, and several private alleles, suggesting that there's a presence of an active sexual state in Malaysia. These results were very surprising since they contradict what is known for the older populations, as well as for closely related pathogens, all of which appears to only reproduce asexually. Sexual-driven evolution of destructants in Malaysia is concerning because it may lead to a more adaptable pathogen that is less responsive to disease management practices. So this brings me to the question, what is the origin of destructants? Native populations of fungal pathogens typically have higher genetic diversities compared to their introduced counterparts. This is because introduced populations usually emerge from a small number of populations, where native populations have had long periods of time to evolve. Does this mean that Stratosphere destructans is native to Malaysia? No, this is very unlikely because Malaysia does not have any eucalyptus species native to the area, and Stratosphere destructans is very specific to eucalyptus. The Malaysian industry is also extremely young, where their first eucalyptus plantation was only established in 2008. This indicates that all of our investigated populations represents non-native populations, and the origin of this pathogen remains unknown. It is, however, expected to be either Australia or East Timor. The reason for this is because the majority of eucalyptus species are native to Australia, and other Stratosphere foliar species have been discovered in non-native populations before they were finally observed within their native range. And then for East Timor, Stratosphere destructans has been reported on one of the native eucalyptus species in East Timor. So we found strong geographical structure which suggests independent introductions into all of our sampled regions from an unknown source. However, two of our earliest collected isolates from North Sumatra shared a genotype with isolates from Thailand, Vietnam, and South Africa, and it seems probable that North Sumatra would have been the source of disease outbreak for at least one of these countries. Then North and South Sumatra were found to be genetically closely related, therefore destructans was probably introduced into South Sumatra from North Sumatra. Then the South African population was clonal, even though the collection consisted of isolates from multiple sampling sites. This indicates a single introduction of this pathogen into South Africa. So our final aim was to assess the microsatellites as a potential diagnostic marker. Identification of Stratosphere foliar species are time-consuming and expensive, and this is because morphological characteristics and initial disease symptoms of both the aggressive and the less aggressive Stratosphere species are very similar. Then this genus grows very slowly in culture, and as you can see on that picture, we had less than 2 centimeters of fungal growth after 5 weeks, which significantly delays the DNA extraction process and as well as the identification. Then once we have quality DNA, identification to species level is not possible based on ITS, and we require at least 3 fungal barcoding gene regions. So due to the time and cost involved, large-scale studies typically sequence only a subset of isolates, which leads to inaccuracies. So we need a rapid and cost-effective tool. So we used our developed microsatellite markers from Stratosphere destructans, as well as one newly developed Stratosphere epicoides microsatellite marker. And this was used to distinguish among 6 Stratosphere species. Why would we want to use microsatellites? Because it's got a high amplification success from very low quality and quantity DNA. And we can look at 13 loci in only 2 PCRs. So this is a snapshot of our results. Highlighted in the table are the specific loci that did not amplify for each of our species. And based on the size of the amplicons, the microsatellites can accurately distinguish among our 6 stratosphere species and can be used as a high throughput and cost-effective diagnostic tool, especially in large-scale disease surveys and inoculation studies. I would like to thank everybody that is involved with this project, especially our funding bodies, for their invaluable contribution. Then I would like to thank you for listening. And if you have any more questions, I will be available for afterwards, and you can also read our publications. Thank you so much.
Thank you, Minette. This was a very interesting, very laden talk. But I can see Pedro has already got a question on the sexual structures. So he's asking, have you been in Malaysia and have been sorting through leaf litter? But I guess Mike should answer that question.
I can also answer the question, Salon. So I think you can hear me. So I don't think we've been back to Malaysia since the initial sampling, but I know Mike has been sampling leaflets in Indonesia before we discovered that this pathogen seems to reproduce sexually in Malaysia. Our best second bet was Indonesia. So Mike did go sample for leaf litter, but it is very difficult to find because there's so many different fruiting structures in the leaf litter. So maybe if we go back, it might be then. Yeah.
So, Minette, I don't see, yeah, I see another question. Bruno wants to ask another question.
Annette, thank you for a nice talk. I'm wondering, with pathogens such as this, we view them as eucalyptus pathogens, but especially when, you're seeing these very clonal populations, what are the chances that this thing is actually, exist on some other host? And what we're looking at are the populations that have jumped and become, have caused these big outbreaks, that there might actually be another primary or alternative host.
Yeah, that's a very interesting question. I think it's very unlikely because all of the Stratosphere foliar species, or especially the very pathogenic ones, has only been found on eucalyptus species, not even really Corymbia, which is very closely related. And Stratosphere destructans specifically is very host specific to only a few eucalyptus species. It's not even occurring on our temperate species. So the chances are very slim that it's on a different host and it host jumped. I know it has worked with rust fungus, but it's very unlikely.
Thank you.
Yeah.
Thank you, Minette. Thank you to all the speakers in this part of the SASP 2021. We will have a 10-minute break now, and we will resume at 12:30. Thank you.
Thanks, everybody.
Okay, break is over. We'll continue with the next speaker, who is the only male speaker in this session, Jan Nagel. He's from the University of Pretoria and he did his PhD in genetics. And he is most interested in looking at what genomics can do in order to solve interesting problems. And his talk today will focus on the Botryosphaeriaceae.
Good morning, ladies and gentlemen. I will be presenting on the development of highly transferable microsatellite markers for the Lasiodiplodia and Neophysicoccum. This work was published last year in Fungal Ecology. The Botryosphaeriaceae is a large family of Dithydiomycetes and contain many prominent genera such as Diplodia, Lasiodiplodia, Botryosphaeria, Macrophyemina, and Neophysicoccum. Species from this family occur mostly on woody plants, but not exclusively. Many economically important trees are affected by diseases caused by species of Botryosphaeriaceae, such as fruit trees, plantation trees, but also crop plants such as soybean and wheat. These fungi are also commonly isolated from trees in their native environments. There is also a large variability in the observed host ranges of Botryosphaeriaceae species. Some species have very large host ranges such as Botryosphaeria dithydia, Macrophyemina fasciolina, and Neophysicoccum parvum, whereas other species have much narrower host ranges such as Diplodia serpinia and wheat-associated Euterospirella species. Many species of Botryosphaeriaceae are able to cause asymptomatic infections, where they are able to infect and persist in the host plant without causing any disease symptoms. Many of these species are thus frequently isolated as endophytes. However, environmental stress can trigger these fungi to become pathogenic. Such stressors include extreme temperatures, physical damage, and drought. Given the growing number of invasive plant pathogens across the world, it is becoming increasingly important to be able to study the population dynamics of these organisms, but few robust genetic markers were previously available for the Botryosphaeriaceae. Microsatellite or SSR markers have previously been designed for the most prominent species, but these have rarely been tested for their transferability to other closely related species. The availability of genome sequences of multiple species of a genus are extremely beneficial for marker development, as it allows one to design primers that can amplify in target species, but also with a high likelihood of being amplifiable in other species of the genus. At the onset of this study, we had genome sequences of multiple species in the Lasiodiplodia and Neophysicoccum. Our aims were thus to develop SSR markers for Lasiodiplodia and Neophysicoccum using the available genome sequences, to determine the transferability of these markers to other species of these genera, and to demonstrate the utility of these markers within test populations. Our strategy was to use a collection of de novo and publicly available genomes to identify SSR containing loci common among species of each genus and to develop markers from these loci. We used Sirocco to identify and characterize the SSR containing loci from these genomes. These were compared between species, and primers were designed and screened using various tools from CLC Bio main workbench and Primer 3. Selected primers were tested using PCR and gel electrophoresis. Lastly, we assessed the level of polymorphism for selected markers through the analysis of labeled amplification fragments using GeneScan and GeneMapper. We designed primers for Lasiodiplodia and Neophysicoccum species respectively. For the Lasiodiplodia primers, we designed 3 sets of primers. The first using all 3 available species genome sequences, the second using the genomes of L. pseudotheobromii and L. theobromii, and the third set using only L. theobromii genome sequences. 2 sets of Neophysicoccum primers were also created. the first using the genomes of 4 species of new Physicoccum, and the second set using the genome sequences of 4 N. parvum isolates. Many PCRs and gels later, we determined the transferability of markers to other species in each genus. For example, in the electropherogram above, we considered 4 markers. For the first marker, all isolates with successful amplification had single bands. For the second marker, many species presented additional amplification products. Markers were deemed transferable to a specific species if they resulted in a single amplification product. Some markers, of course, failed in all species and were excluded entirely from the set. 77 out of 92 Lasiodiplodia markers and 32 out of 43 new Physicoccum markers fit this criteria. The number of markers that successfully amplified in each species are represented in these 2 tables. From this, it is clear that a large number of markers were transferable to species not included in the design process. We did, however, not determine whether each marker is polymorphic in each separate species. We did, however, determine that a subset of these primers were indeed polymorphic in a population of Lasiodiplosia marjangana and new Physicoccum parvum. Markers were chosen based on agarose gel electrophoresis size polymorphisms. From 20 isolates of each population, we could detect between 2 to 7 alleles per locus in the Lasiodiplodia marjangana population and 2 to 5 alleles per locus in the new Physicoccum parvum population. The calculated heterozygosity of these markers varied considerably, but these do not reflect the effectiveness of the marker but are influenced by the distribution of alleles in each population. Combined, these markers could identify a total of 54 alleles and 16 multilocus genotypes in the Lasiodiplosia marjangana population, and 35 alleles and 15 multilocus genotypes in the new Physicoccum parvum population, thus exemplifying the applicability of these primers for population genetic studies. In summary, we developed the largest collection of transferable SSR markers for the use in the Botrys. These markers can amplify in many species of Lasiodiplodia and new Physicoccum. However, the presence of the SSR and the polymorphism need to be confirmed for each species and population. The application of these markers will allow populations of many of these species, especially those lacking genomic information, to be studied. I would like to thank our funding bodies for making this research possible and allowing us to keep plants healthy.
Thank you, Jan, for a very interesting presentation. Does anybody have a question for Jan? But I can start, Jan. So, how are you going to use those markers now in the next phase?
So at the moment we don't have any specific plans yet, but there are some studies, for instance, I think some of LC Kruiwagen studies that already use some of these primers, and then Joseph Macquarie, they were my 2 collaborators on this study. They already are planning to use some of these markers, but further than that in the future, not yet any further plans.
Any other questions from the audience, from the floor here?
Maybe I can add a question to the end of time.
Yes.
I'd like to add a question for Jan. Jan, so how is this changing? You know, I'm thinking about other Botrys species that you haven't tested within this group. You know, how might this change the way or people's ability to do population genetics with this, use what you produced here and possibly apply it much more broadly on species that you haven't even tested yet? Do you think that's likely? possible?
Yes, I think so, because one of the big problems in the past, as I stated in the introduction, was that many of the primers that were previously developed wasn't tested for how wide they're able to amplify. And with these markers, they are able to amplify a much wider range. So it gives the person that wants to do population genetic studies a much wider set of markers to choose from, to investigate whether there are polymorphisms, to choose markers that will actually be informative for his population.
Thank you.
Okay, thank you, Jan. Our next speaker is Lisa Rothman, and she is an epidemiologist from the University of the Free State. She's very, very passionate about her work, and she goes to the field quite often, as I found through my correspondence with her. And she has brought some really new methods for analyzing epidemiological data from Brazil. And today she will be talking about how she applies these new methods to epidemiology in plant disease of Sclerotinia sclerotiorum.
Good day from a sunny Free State, and thank you very much for the opportunity to share a portion of my PhD research and a future publication that I'm going to be presenting on quantifying a decade-long survey of Sclerotinia using reproducible research tools. So in the beginning, I did not have a great relationship with R. I knew it existed. I had no idea where to start with R statistics. And in 2018, I met Professor Emerson Del Ponte at ICPP in Boston. We sat together for 12 minutes, and that 12 minutes changed my entire life. I went to visit him in 2019. Last year, I finished my PhD, and today I am presenting this talk to you on using reproducible research tools, being excited about our studio and other reproducible tools to do our research and to communicate our research. So the aim of my project was to quantify spatial, temporal, and potential interactions that were occurring in the data, and ultimately also to share this, not just a paper or a presentation, but to share the data, because sometimes fresh eyes can give a whole new perspective on what is hidden within data. So the Crop Estimates Committee conducted a survey with soybean and sunflower producers, and this was done by telephone, email, and through Grain SA newsletters. And the producers responded back to the Crop Estimates Committee, and this was repeated for 10 years from 2006 to 2015. The survey collected the area that the producer planted, the amount of, or the hectares that they thought was infected with Sclerotinia, as well as the previous planted crop from 2008. So these are just some symptoms that they had to work with, which were in a written form, and there were potential limitations associated with this. So the GPS points for the farms were not necessarily taken, but for the town as their nearest locality. There were unbalanced responses between the years as well as the locations. Agronomic information was not necessarily collected across all the years. So if it was irrigated or not, what was the previous crop or not, was not collected each year. The correct diagnosis was also assumed, and inoculum presence was not noted, whether there were apothecia, ascospores, or mycelial inoculum was not necessarily reported. So I also thought it was important to look at, does the sample size of the survey represent reality? And how can we go about this? Well, I first had to start by organizing the data and organizing the findings before I could really get into even exploring the data. And I'm sure many of you have experienced a similar situation where you save a file and you give it a new name and you give it a date, and it's version 1, version 2, version 3, version 3 seen by professor, version 3 seen by professor, returned to student. And that made me really doubt the work I was doing, because after a while I couldn't really track what I was doing, and there was just so many data points, and yeah. I then got introduced to Google Sheets, and this allowed a wonderful version tracking environment for me. And it's also really good for teams, because if you are more than one person working on the data, you can see who's added or changed something over time. Where did I start? So when I went to visit Emerson and his students in Viçosa, Brazil, I started with using R for data science. And I looked at Hadley Wickham's Tidyverse, and I used that as my base for learning R. And I went from the importation of the data all the way to communication through statistical programming. And I use the language R. I use a graphical user interface, which makes it much user-friendly for someone that has never coded before, like using RStudio. And all my version control was done with Git and GitHub. Now, over these 10 years, as you can see, there were different names and cases assigned to all of the cropped data. So if you had to go and do this manually, you'd have to change each of these manually. But using R, there was an automated way to ensure that all of the responses were correctly reported. You can also see that there's some dry land and irrigation reports, as well as one report that did not have a year assigned. So I love Hadley Wickham's work on the Tidyverse. And he said that tidy data are a standardized way of linking your dataset's physical layout with its meaning. And in the beginning, I had a really untidy workbench. It was chaos. And learning to use the Tidyverse, I actually managed to produce something that had structure and had meaning. And this allowed me to see, all right, in 2007, they reported some irrigation and dryland crops for both soybean and sunflower. And now I have two separate data frames that I can work with. And this is fantastic, because now you can continue with confidence. So it's not rubbish in, rubbish out. So what are the variables I extracted? Producers reported the infected area as well as the area they planted. And I made this a percentage, and this became the intensity. Then there was the number of positive responses divided by the total responses. I made this a percentage of the Sclerotinia reports, and this was my prevalence. And then you have the area reported in the survey and the South African area planted as estimated by the Crop Estimates Committee. And this then allowed for the extent of the survey to be recorded. So using a visualization in the past, I would have used Excel and Microsoft Office. Now, there's nothing wrong with this graph at all. I've made many of these in the past. And they have the standard error bars, but maybe my summary statistics are actually hiding something quite interesting. So I thought, why not integrate intensity and prevalence in a graph together? This way, you can present the mean, which is what this bar is presenting, the confidence intervals, as well as all the individual points reported. And these individual points reported allow us to make a prevalence bar, which is this shaded bar here. Now we can see that in a year like 2006, a lot of the reports were positive reports, whereas in 2015, the majority of the reports were negatives, or not negatives, no disease, in that sense negative. And that's why it has a low mean as well as a low prevalence. In the past, I would have visualized my maps using screenshots from Google Maps and pasting them in Microsoft and Paint and trying to get them to work. So if I had to recreate this today, there's no way I could. I mean, this is usable. I presented this at a poster at the National Sclerotinia Initiative. So it can be done. But there are better ways to do it. And for me, and that's what made me feel more confident in the work I was doing. And using the R Natural Earth package, I could go and develop a 10-year overview of what was happening of the binary responses. So was the disease present or absent? And this allowed me ultimately also to make a time lapse of what was happening over the years. And while I was doing this, I got this new idea, thinking, well, do these responses even represent reality for what was happening in the area that, or the extent of the survey. So I decided I should go and look at what is the area planted to soybean and sunflower in South Africa, divided, take that sample that was reported in the survey and go and have a look. And this allowed me to really make a true or false statement on the area planted. So for 2007, it was not, most of the provinces were not representative of the area planted to the crop. But in 2012, for example, all of the provinces had a high enough percentage of extent, so what was reported in the survey could represent reality of what was planted in South Africa. So this gives me more confidence in the work I'm doing, as well as when I report the data, producers and researchers and industry members can go and see, all right, in reality, there may have been a lot of sclerotinia occurring in 2009, but I should be very careful of how I interpret the data for the Northwest province, because there was only one response or one mean value for the Northwest. So this will not indicate the whole of the Northwest province. Another example, for example, may be in 2012, we can know that these responses are all more reliable. But how does this work if we look at prevalence? So in 2009, we see that there was only one positive response in Limpopo province over here, and it was a very high prevalence. So the mean for this town was much higher than, for example, over here in Mpumalanga. So in 2012, it was representative of reality that there was not many observed sclerotinia reports, but you wouldn't go and say that, we can now reliably say there was no sclerotinia in the Free State. We can just go back and look at, was the area representative or not? Another thing that I really love about our world is the fact that there are many packages available, and some packages actually include data that are from other sources. So Adam Sparks developed a NASA Power package that includes NASA Power gridded weather data that's simulated. And we went and I went to take ground truth weather data from local weather stations, and I did a regression or correlation analysis with NASA Power data, which is at 2 meters above the ground level. So this is quite nice and close to the area that I would be interested in for soybean and sunflower. And it is a significant p-value, and it has a high R square or R value. So for the purpose of our work, this is sufficient enough, because we could go and correlate the temperatures, the mean temperatures for April, February, January, March, and with our mean sclerotinia intensities, and see, are there any potential parameters that stand out? So another statistical tool that I used within R, which is base R, was Mann-Whitney test. And some of the producers, not all of them, provided which was the previous crop planted. So we focused on, I'm going to focus for this example on maize and soybean, and there were only 210 responses out of the 1,300 approximate total responses made. So the medium intensity when maize was planted in the previous season was about 8%, and the median intensity was about 14.5% when it was a host crop in the previous season. And using the Mann-Whitney test, we can say that there was no significant difference between these, having them in the previous season, which shows that this data set says that if you have planted a host crop or maize in the previous season, it's not going to influence sclerotinia in the following season, which aligns with what other reports or other literature has said, that rotations do not necessarily provide enough protection against the pathogen. So where does all our time go as researchers? And this is actually what my talk's really about. It's about how we can use reproducible research tools to gain some of our own power back and our own time back. I did a quick survey on Twitter, about 36 respondents, and a majority of the respondents, over 40%, said they spend more than 7 hours a week on admin. I'm sure many of you also have similar experiences. So I thought, why all the effort? Is it even going to make sense for me to learn a new skill? And I really don't have enough time. Where am I going to find the time to learn a new skill? What's really great about learning R and R was the fact that I could go and trace all my steps. I could provide a review for someone to see if I'm doing my science to the best of my ability. It can be citable. So not only are you having a publication, but you have citable code. You can even put your preprints in somewhere like Open Science Framework, where people can cite the work before a preprint. So this is just something that I'm working towards in my lab. I started learning R when I was, last year I turned 30, and that's the mean age for starting to learn R. So I know that there's a lot of like, but I'm too old to learn R, or I'm too advanced in my studies to learn R. And that's not the truth. This is just a survey, a small survey that was done with the R community survey. And a lot of people end up enjoying the skill that they've learned. Most of the highest or level of school that they've completed, that have started learning R, is masters and doctorates holders. So there really is the space for you to learn still. And I started doing this because I wanted to track my progress and get more confidence in the work I was doing. I used additional tools like gradient machine boosting, random forest, and logistic regression modeling for the other chapters of my PhD. Some of you might be interested in how R and RStudio can be used to analyze your genomics data, or even just organize and structure it. So another thing is you can learn heuristically, have a heuristic learning process through detecting errors using R in your data. And I learned most of this by myself, being supported and surrounded by people that use R. I know that it might take months or years to become proficient, but that's what we have a community for. And having a sense of belonging at Open Plant Pathology has really helped me with my errors. Yes, language, syntax, and error messages can be very frustrating, but it can also be exciting because you will learn to communicate with peers and colleagues from around the world. And ultimately, people keep coming back to R and RStudio because the community is available for us. And there are packages that are really flexible, and it allows a lot of work to be done. So I'm really grateful to everyone that contributes to R and RStudio. So thank you for letting us stand on the shoulders of giants. I'd like to say thanks to all my supervisors and the peers that have helped me, and everyone that could support the research being put out today. You can find a citation, a copy of my presentation, and I'm looking forward to any questions. So thank you for your time, and I hope you enjoy the rest of the conference.
You need to step again.
Apologies, Lisa. We just needed to unmute there. Thank you for an interesting talk. Lots of things to think about there. I must open by congratulating you also, thanking you for your lively tweeting. Very, very effective. So thanks a lot for that. And thanks for reinforcing, taking us on a journey that you've been on with R and recommending that to others as well. Let me see if there are already questions in the chat box. Yeah. I don't see any questions there. Lisa, perhaps to sclerotinia, a question about, this is one of the most serious diseases in South Africa for our farmers. What are the next steps? Where are the big gaps in our knowledge in terms to support our farmers to deal with this serious disease more effectively?
So thanks for the question. I hope that everyone can hear me. Great. So I think something that really needs to be looked into specifically on sunflower is the fact that there are no products that are registered to protect the sunflower against sclerotinia. There are registered products for soybean and canola, as well as pea. But I think that there's a big gap when it comes to sunflower, especially because the risk is so much more, considering, soybean has indeterminate growers, but a sunflower, not necessarily, not at all. And our producers are also, reliant on, when they see sclerotinia in the field, it's usually too late. So when do we apply these fungicides? But it doesn't help we have a forecast, but there are no products for them to apply. So that's something that I think industry also needs to get involved in, actively looking at what is available and what works internationally. And yeah, I think that's something that, in my opinion, could contribute to curbing the persistence of sclerotia in soil.
Thanks, Lisa. I see more comments in the chat box thanking you for supporting and motivating people to learn R. There's a question here, say, where would you recommend a student begin to learn to use R? From Megan.
So I started at the very deep end. I already had everything on Excel, and I was like, okay, I need to find a better way to do this. And thankfully, Emerson, we used our own data set. I see you asked, should you use your own data set or scripts from somewhere else? There are a lot of scripts available. Something like Tidy Tuesday. A lot of people publish their work that they've done from Tidy Tuesday, which this week is like the Kenyan census. So you can go and replicate or reproduce what someone else has done. And if you run through every step of the code with what they've done, you'll actually learn, what does it mean? What are you actually doing? And that's what I loved about R the most, because I wasn't just regurgitating what someone told me to do. I was actively learning. So I can send out, I'll tweet some more resources and great places to learn, but I would just say start. Don't be afraid of R. There's a really great community, and maybe I'll take Sofien up on his challenge of starting an R community class.
Yeah, thanks for that. I'm going to stop it there, but thank you again for that, and for also demonstrating and living up some of the things that Sofien was recommending this morning, being open and sharing and getting others involved. So Lisa, thank you very much for the talk. In the meantime, my link to the meeting has dropped, but you can still hear me through the central speakers. So apologies for that you can't see me at the moment. I'm going to pass over to Mike Wingfield for the next talk. We're very sorry that Teresa Coutinho could not be with us. She would have done this talk, but unfortunately is sick today, and Mike has agreed to do it. It's appropriate that he does so because he and Teresa has worked very closely to put together a book on the history of plant pathology in South Africa. Sofien said this morning, for bad things to happen, good people need to do nothing. And this is one of those tasks that really could have fallen by the wayside, but passionate people have taken it up, and we're very thankful that they did. So Mike, if I can pass over to you to introduce us to the book.
Philip, please, if you'll just share the screen for me, please, Tom.
I can see you. I can see you from
You can see me. So hello, everybody. It's great to have a chance to greet all of our members of the SASPP and other friends from other parts of the world. Bernard has already said that Teresa was to make this presentation, and I must admit that I'm very sad, and she's online, so she can listen to me. Teresa, you can hear what I'm saying, that she's not here to do this job, because Teresa has really done the heavy lifting, the really heavy work for more than a year in putting together this very important book on the history of plant pathology in South Africa and the history of our society. Teresa got struck down, if you're curious, by a viral disease, and the viral disease is shingles. It's very horrible. I can tell you all that from personal experience because I'm busy recovering from shingles myself. And if you haven't had a chickenpox vaccination, if you're an adult, go and have it, even if you had chickenpox, and it will reduce your chances of being infected with shingles. It's worth trying. It's really a horrible problem to have to deal with. So, Teresa, I hope that I can do your talk justice. I'd like to take the opportunity to thank the organizing committee of this meeting for giving us the opportunity to say a few words about the book, The History of Plant Pathology in South Africa. I'm not going to talk formally in any way at all. I'm certainly not going to cut through the small detail of the book, The book in text is about 370 pages. The final printing is not done, so it's a very extensive piece of work, and you don't want to hear all of that detail. But I will show a couple of slides. Let me see if I can forward to a cartoon. Cartoon will help me. Not. There we go. Yep. Down arrow, not right arrow. So let me just start quickly by explaining why this book was put together. So Teresa was the president of the SASPP previously, and at an AGM of the SASPP in 2019, as you can see, she, not me, she agreed to put together a history of the society. There had been a number of other short histories put together. John Mildenhall put together some pages, a small number of pages. Chris Riley did the same, but the job was never comprehensively done. So she agreed to the task, I think very bravely. But it was also, I think, driven strongly by her, and subsequently my joining this party, curiosity about the history of our discipline in South Africa. And we also believed that this was a good way of celebrating 130 years of plant pathology in South Africa, over 50 years of the SASPP, or SASPP congresses, and then, of course, in the International Year of Plant Health. We had hoped to come to this meeting with the final copy of the book. The book is with publishers at the moment, and for those of you who have dealt with publishers, you will know that this is quite a tiresome process, and will probably be another few months before we have a hard copy, and then we'll share with all of you that are interested the information on our website. And Lisa can tweet, and others can tweet too. So let me just say a few words about, I'll tell you perhaps a few stories about our experience with this book. We made many discoveries, and Teresa and I drank many cups of coffee together, mostly in social distance, starting with the contents page and then trying to thrash out detail. It was fascinating. We had wonderful discussions and we made many discoveries. I mean, from my own perspective, Teresa would say the same thing. I learned how incredible people were of the past. So the early days in the late 1800s of plant pathology, people did the most remarkable work under incredibly tough circumstances, much tougher than some of the circumstances that people complain about today. Very little money. What they had is a passion for the subject. They believed that plant pathology and plant health was crucially important, and they just did amazing work in various parts of the country. We discovered that plant pathology, if you're looking for a starting date, plant pathology started in about 1887. So there were two papers. One was concerning diseases of apple and pear, and the other, now this is close to my heart, was concerning some diseases of trees. So the history of plant pathology started about then. It was the first recognition of plant diseases in the country. So those were the first two articles. As we started to put this book together, we were mindful of the fact that the initial promise was to put together a history of the society. But that changed. It changed for a number of reasons. We became fascinated for one point, on one side. The other was one plant pathologist in South Africa, as we started to ask people to write pieces on plant pathology at universities, plant pathology relating to particular crops, and many other related topics. One person in particular wrote an amazing piece, and this was Carrie Peterson, Professor Carrie Peterson, who, as many of you know, is a plant virologist. And Carrie, I think you're online, and thank you so much for this. Carrie wrote more than 30 pages capturing the history of plant virology in South Africa. And when you see the book, please take that piece and read it, because it's absolutely amazing. It's highly detailed. But what we realized is that there were similar sorts of stories. Some would be longer, some would be shorter, but others would contribute. So we then started to accumulate information relating to the history of plant pathology in South Africa. We asked a special friend, and I was hoping he would be online, and it's Professor Fritz Reichenberg, one of my early mentors, one of Teresa's early mentors, to write a preface. So Fritz was faced with two tasks. The first was when we asked him to do this, the idea was a preface to a book on the history of the society. And Fritz had spent a lot of time, including time here in FABI, digging through boxes of the archives of the society. So he was the appropriate person to do this. But later we had to ask him to change what he had written to accommodate more than just the society history, but history of plant pathology in South Africa. And he's done a wonderful job. And Fritz, if you're not online, you will receive the message from others. That, we appreciate this very much. And all your colleagues, all plant pathologists in this country and globally, are exactly the same. So the book really has two parts. It captures the history of plant pathology as best we could do this. Pieces written by many of you, and thank you all for this. And there's an acknowledgement up front as to who wrote what. I mean, we've edited very deeply. We, in this book, say we take personal responsibility for cutting and pasting and editing, and we hope we'll be forgiven for all of that. So it's just the only way we could do this. And obviously, because different people wrote these initial drafts of pieces, we wrote a lot of it ourselves, the style changes through the book to some extent. But it comes together, I think, relatively nicely. So two parts, the history of plant pathology in South Africa, and then a focus on the SASPP. So another discovery, maybe an issue highlighted, and I want to here address particularly young members of the society. And that is because older members of the society, I'm one of them, some of the early members of the society are moving away. And what we realized is that societies, or what came to mind, is how important societies are to keeping a community of scientists together. I'm absolutely convinced that without the SASPP, we would not have our community bound together the way it is today. The fact that we have a meeting such as this, and Teresa and I wrote in the special issues, so Professor Jane Carolus will address you shortly, but we wrote, and I hope you will take the time to read that, a short commentary on the history of the society. That has quite a lot of detail in it, but it's summarized into a short commentary, as opposed to in the book where there's a lot more detail. For those of you who are curious, the society is about 60 years old now, and the society was started by microbiologists and plant pathologists, a small group of them. Chris Rabi, previously of the CSIR, and previously before that, I think the MRC, got a small group of plant pathologists, microbiologists together. So the microbiologists interested in plant diseases, Ben Stradham was another, to start a society. That goes back to 1962. And off it went very successfully as a society that captured both microbiology and plant pathology. About the 1970s, things started to get a little bit complicated. The microbiologists were starting to focus, not all of them of course, but on other aspects of microbiology, medical microbiology and so forth. So a tension started to emerge between these two groups. I was a young plant pathologist in those days, and I remember that tension, looking at it as a student and wondering what was really going on. There was quite a lot of animosity, and in fact the society meetings, a meeting like this, were sort of separated into two pieces, microbiology piece and the plant pathology piece. And some of the microbiologists that worked on plant diseases caused by say bacteria weren't sure where to go. So this was very complicated. And then in 1980 the conflict became quite tense. I was at that meeting in 1980, and we talk a little bit about it in the SAJSI article. It wasn't nice to watch from the perspective of a young plant pathologist. I was quite shocked at the things that were said by people to other people in this little room on the campus of the University of the Free State. And I won't go into those details, those can perhaps be forgotten, but the society split, and therefore we had the SASPP, which was the South African later, the Southern African Society of Plant Pathology, and SASM, the Microbiology Society. There was fear that our society would actually survive. Maybe the group was very small. It was suggested that we would collapse. In actual fact, the society has done incredibly well, and it's grown from strength to strength. And again, to the young plant pathologist, this is your society. You have to take leadership. You have to take leadership from the old guard, us old people, and start to run the society and to take care of the society with every bit of energy you possibly have. Just heard Lisa talk with tremendous pathogen. It's that tremendous passion about pathogens. It's that sort of passion that is going to ensure that the society continues. So let me end. I've spoken for 10 minutes, and I think I said I would speak no more than 15 minutes, but I'll end before that. Let me end by sharing just a few slides with you and some pictures. They're just pictures. So for those of you who are not South African and haven't been to one of our society meetings, the next one will be hopefully in person, I'm sure in person here at the University of Pretoria in our wonderful facility, Future Africa. And you'll be able to very quickly get to game parks and other wonderful parks. So this is just a picture of some of the Congress venues. I won't go through it, but we have the privilege of moving around the country and having meetings in beautiful parts of the world. They're great meetings, and maybe our next will also have a bit of a hybrid feel to it, so we will also have friends from other parts of the world attending. We have always had keynote lectures. Teresa has chosen these slides. There are more people than these. I'm just looking and seeing who I can remember. Art Kellerman, John Lucas, Noel Keane, Jim Cook, George Carroll, Bryce Kendrick, Mary Palm, Carolee Bull, Lindsay DeTroy, who is on this call. I think, Lindsay, great to see you, and thank you for the incredible support that you give our society. You're very special to us. You're the APS president, but this allows Andre Drent, Michael Mulgrew, and Ian Toth, and others. So we have the privilege of having had wonderful keynote presentations, and today we witnessed another with Sofian presenting a beautiful talk with us. And we're hoping that Sofian will be here in person next year for our meeting. And Sofian, then we will take you to see some other parts of South Africa. The society, we have memorial lectures, particularly important, the Van der Planck lecture and the Deutsch lectures, and some of the Van der Planck lecturers. And again, it's just some in pictures here. Fritz Reichenberg and Zach Victorius and Neil McLaren, Mark Lang, Chrissy Ray, who's sitting at the back here. Chrissy, it's a nice picture of you. Officer Carrie Peterson, who I mentioned earlier, Chris Coppers, and Landgraf, Joanna Damas, Marco Kreisner, Kat Maria, Pedro Kraus, and Sandra Denman. Hey, Teresa, you let me out, so I'm sure I get one of these pictures. But anyway, others as well. I think everybody knows who Van der Planck was, perhaps the most famous plant pathologist ever to have lived anywhere in the world. And Teresa and I, with Andre Drent, put together a commentary in that special issue of South African Journal of Science. And that was an interesting experience for us as well on Van der Planck. So please read that. And those of you who teach students, let them read and learn more about a very, very interesting plant pathologist in South Africa. Honours and awards. Honours awards were slowly introduced into the society. Now we have a whole lot of them. One will be presented by Venant today. Numerous, so that's the John Peterkin Morton Hall Award. Pictures here of recipients of the Hussoon Medal. The first recipient was Van der Planck, and there have been 5 since then, I think. One about every 10 years. I don't, people in the room here are laughing looking at this picture. So that is John Milton Hall. He is doing what is called the Milton Hall Stakes. It's other names as well. So this is, for those of you who are not South African, South Africans all know about this, well, maybe not our young members. So this is John Milton Hall. The story is on our website. I think it's on the FABI website. Right? We must go and check that. Certainly was at the time, and there's a demonstration of how to do this. So should all go and try. The pictures of me trying, but I gave up many decades ago. But John, I don't know how old John was when he was doing this, but at least in his late 60s. The point is put a beer bottle on your head, and then you need to drink the contents of the beer without touching the bottle. It's a piece of amazing gymnastics. So I watched this as a young plant pathologist for years, and then I remember the first year, other people, 3 young plant pathology students from the University of Natal and Peter Marisburg, did this very quickly, and I won't talk about that, but it was an interesting experience. Since then, many people try, and some are successful. But the Milton Hall Stakes is now just a part of the what we do at our society. John, I think, has stopped. I think he told me he has stopped. We have wonderful congresses full of fun. Here you see some of us enjoying some of our plant pathologists enjoying themselves in all sorts of crazy ways. There were some more explicit photographs that I asked Teresa to take out. I think that picture with the togas was the first Club Mechanos conference, and I won't go into any details about that, but there you see Chris Rabi and I see Francois Wolfart showing a leg. I can't see who else is there. So we have wonderful conferences. The parties are special. So in conclusion, I have taken exactly 15 minutes. I need to stop. I hope that you will enjoy seeing this book. We talked a lot about how to publish it. We want it to be available to everybody, so it's to be free. So it will be published free. So we're paying, or we raise some money, I won't go into those details, to do the publishing. Obviously we need to pay the publishers for layout and so forth. And then it will go free on the SASPP website, and anybody that contacts us will receive it free. We see it as a work in progress. So for sure it's got its warts and bumps, and we will be adding to that, and maybe others will take over. But it has formed, without any question, a foundation for collecting and making sure that the history of our society and our discipline is there for posterity. So work in progress. We hope you'll enjoy it. We will print some copies to give to those that contributed to the exercise, and perhaps some others and a few libraries. But there won't be enough, unless we have enough requests to print more, and then we'll have to talk about how to fund that. But in conclusion, to all of you, in our final discussions in the last few weeks, our coffee party last week, in fact, Teresa and I thought that it would be appropriate to dedicate this book to Terry Aveling. So there is Terry. Terry was I met Terry as a young plant pathology student. She was first in Maritzburg with Teresa, in fact, at that time. And she was a stalwart plant pathologist throughout her entire career. She's a wonderful plant pathologist, a wonderful person. I believe that she would have loved this book. The book is dedicated to her, and we will make sure that it also then goes to her loved ones and her family. So thank you all for listening. It's really great to have the opportunity to talk with you. And finally, my role is to welcome Professor Jane Carruthers. Jane is I think emeritus professor, is Jane, hiding behind me from UNISA. Jane is a wonderful historian, and she, in terms of this particular role, has been the chief editor of the South African Journal of Science. I won't talk about the wonderful support she's given many of us as the chief editor of South African Journal of Science. And one of her initiatives and her goals in the International Year of Plant Health was to produce a special issue of the journal commemorating the International Journal of Plant Health. Commemorating the International Year of Plant Health 2020 in the journal. So Jen, over to you. What I have to do is I have to spray me. I have to spray here. I'll spray you. And I'll hand over to you.
Is it open? I'll just start your presentation.
Did not share the screen yet. Can you share it?
Let's share the screen. We are ready.
Thank you, Mike, for that introduction, and thank you, everybody, for the invitation to come and chat to you very briefly this afternoon about the special issue of the South African Journal of Science. Well, on the 28th of May, 2019, three of the editorial team of the South African Journal of Science, Teresa Coutinho, the Associate Editor for Agriculture and Forestry, Linda Fick, who's the Managing Editor, and I, the Editor-in-Chief, met for coffee in Pretoria. The reason for our getting together, which you could do happily in 2019, was to discuss Teresa's idea for a special issue of the South African Journal of Science to celebrate the International Year of Plant Health in 2020. This, as you all know, I think, is an endeavor of the Food and Agricultural Organization in the United Nations after the 2018 adoption of the UN General Assembly for this special year because it would raise global awareness on how protecting plant health can help end hunger, reduce poverty, protect the environment, and boost economic development. The South African Journal of Science seemed to be an ideal vehicle for this because it has a huge mailing list, it's widely disseminated, and it is extremely interesting going to policymakers, the public, as well as specialists. As we worked out how to get this moving for an issue in the second half of 2020, we became increasingly excited. And there were names of potential contributors, commentaries and reviews, appropriate research topics, and many disciplines flew across the table. By the following day, the three of us were exchanging emails, and even then, working out some of the details for the special issue that you see up on the screen. Just a few days later, Teresa had already sent out invitations to her wish list of contacts. Between Teresa and me, we began contacting the FAO's Mirko Montuore and his team. Linda opened a new workflow on our system, and we enthusiastically set about creating what we believe to be one of the most interesting and important issues of the South African Journal of Science in its very long history since 1905. After a few hiccups, many relating to COVID-19, the special issue was finally published as issue 1112 of 2020. It's freely available, open access, online, and comprising the very best recent scholarship on plant health from the subcontinent. I am so proud that the South African Journal of Science has so wonderfully presented your multidisciplinary field in all its variety, and grateful, very grateful, to the community of plant pathologists for giving us this opportunity to launch it at this meeting. The journal is very honored to have produced this bumper issue on an enormously important field of science. Indeed, so important that the continuation of humanity depends on it. The power of microbial pathogens on humans has been so forcefully demonstrated through COVID in 2020, and we mustn't forget that plant diseases have had a similar impact by decimating human populations because we rely on plants as food. A great deal remains to be learned about plant diseases and insect pests, and we hope that the South African Journal of Science special issue will be a catalyst to further research. It's an extraordinary volume. It shows vision, persistence, and a great deal of hard work. We present a huge range of topics, 66 scholars, many more peer reviewers, and copious reference lists that will be of lasting value. Our most effusive thanks must go to guest editor Teresa Coutinho, her mentees Salmina and Mokeli, the authors of commentaries, book review, review articles, and research articles, as well as the many, many peer reviewers and the production team of Linda Fick, Nadia Krobler, and the Layout Company. We all know that COVID-19 upset 2020 totally. We'd wanted to flight our special issue at some of the events that the UN and the FAO had planned, but most of these were either cancelled or curtailed. So we're very grateful for this chance to let you know all about the special issue of our journal. The South African Journal of Science was first published in 1905. And as a historian of science, I have skimmed many of the early conference proceedings and later research articles. There's no doubt that over the past 116 years, this issue, published last November, is one of the most important of them all, and will go down as a reference point for the future. It's a fount of information and of research, and the many hundreds of literature references will become a research tool and a teaching tool well into the future. The lineup of authors is breathtaking, 66 of them, and they come from 7 South African universities, with Pretoria and FABI, not surprisingly, in the lead. International universities include Australia, Mozambique, and Spain. The organizations and research institutes involved are also a very broad representation. The FAO, the Agricultural Research Council, and groups and associations in South Africa, Mozambique, Nigeria, the Philippines, Taiwan, Tanzania, and Turkey. I'll just give you a sense of the contents of this issue. We have 7 commentaries. Mirko Monturi and his colleagues from the FAO have written a leading article in the front of the journal, as has Mike Wingfield and Teresa, as mentioned earlier, a history of the Southern African Society of Plant Pathology from its inception in 1962. There is a history of the discipline from the mid-1920s in South Africa, a commentary by Bernard Slippers, who's now head of FABI. The importance of plant health to sustainable crops. The value of collections, sometimes those are neglected. A biography of pioneer James van der Planck. And it ends with a discussion about what can happen, and so suddenly, the case of the polyphagous shot-hole borer in South Africa. And something that we really celebrate too, a profile of Mike Wingfield, the National Science and Technology Forum Lifetime Achievement winner in the special animal category, which is the International Year of Plant Health. Congratulations again, Mike. The profile was written by our South African Journal of Science mentee, Salmina Mocheli, and she was responsible for attracting a Spanish academic, Victoria Pastor, to write a book review of 2 significant Springer books on plant health, which I think you may all either know or certainly should know of. We also published 6 review articles, very important review articles, I think, by experts in their fields. We dealt with the hunt for plant viruses, recent research on potato pathogens and food security, coping with potato viruses at a time of climate change, tree health in South Africa, a retrospect but also a prospect, the accomplishments of research on wheat rust, and an overview of managing Fusarium head blight of wheat. And as for the 6 research articles that deal with the following crops, 2 about maize, citrus, bananas, rice, and tomatoes. And in fact, I think we can also be proud of the fact that the contents of the special issue speak to 9 of the 17 Sustainable Development Goals. 0 hunger, good health and well-being, decent work and economic growth, industry, innovation and infrastructure, no inequality, sustainable cities and communities, responsible consumption and production, climate action, and life on land. Exactly a century ago, the first academic plant pathology appointment was made, Paul van der Bijl at the University of Stellenbosch, who became Professor of Plant Pathology and Mycology. In 1926, he contributed an extended article in the South African Journal of Science entitled Landmarks in the Development of the Science of Plant Pathology and of Disease Control. In our special issue, Bernard Slippers takes up the best of his story with the plant disease pyramid, the relevance of the original vision of plant pathology in 2020. It's such a pleasure and honor to attend this 20th meeting of the SASPP and one held in the centenary year of the discipline. And I would like to thank once again everyone involved in this special issue in various ways for their hard work, collegiality, efficiency and friendliness, and to express our appreciation to Teresa for getting the SAJS involved over our coffee date so long ago. Please access the journal, enjoy the read. Thank you very much indeed.
Jane, thank you very much for that explanation and giving that overview of this journal, but of the special issue, but even more so for the passion with which you and Linda and Teresa have driven that. It wasn't that, but I'm sure it must have been difficult on your side, maybe even more difficult on this side, managing reviews and writing and so forth during that difficult year. But what has come out is truly beautiful, and as you say, an incredible contribution that SA Journal of Science is making to this society. So thank you for that, and thank you for a wonderful talk.
Thank you very much, Bernard. And
Thank you.
I'll tell you how much I have learned about your discipline and enjoyed it very much too. So thanks, everyone.
Deeply appreciate it. Thank you, Joe. And that brings us to the end of our program. Other than closing it all down, there are a few things for the SASPP to still complete, and I'm going to hand over to Wynand to deal with the last business. Thank you, Wynand.
Thank you, Bernard. And well, what a historic day and fitting tribute to plant pathology in South Africa. And I think this next item is obviously quite a historic event too, somewhat belated. It is the awarding of the John and Peter Mildenhall PhD Award. And you saw John Mildenhall doing his trick. Some of the slides Mike showed. John is a long-time member of the SASPP, much revered, and this was the brainchild of him and his wife, Peter. Upon retirement, John donated a substantial amount of money to the SASPP, from which he wanted a cash award to be made to a PhD student who obtained a PhD that was of practical importance. And this PhD had to be obtained in the year immediately preceding the biannual conference or the awarding of the award. So the SASPP Council facilitated the judgment process, and applications from prospective candidates were received, and there were certain set criteria which are set out on the SASPP website if you are interested. We don't want to go into too much detail today. So the judgment of the nominees was performed by a selected panel that included highly experienced academics and members from the agricultural and plant health industry. So the judges were required to take into consideration the quality of the research and the research values of Professor Mildenhall, which strongly reflect the implementation of practical and applied plant pathology. He had very strong feelings about this. So the recipient of the John and Peter Mildenhall Award will in future be announced at the biannual conference of the SASPP, which would have been this month in person, and the first time, therefore, that this award is now being implemented and announced. So after due consideration, the research of 4 candidates from 3 universities in South Africa were considered, and the judges were unanimous that the work of all 4 candidates was of a very, very high standard. More information will be published on the website in due course. So, with due consideration of the wishes of Professor John Mildenhall in terms of practicality and in plant pathology, therefore led to the award being bestowed on Dr. Megan van Dijk from the University of Stellenbosch under the supervision of Professor Francois Halien. The title of Megan's thesis was Characterization, Epidemiology and Management of Olive Trunk Disease Pathogens in South Africa. We hope you are online, Megan. Congratulations. This is a truly historic award from the context of plant pathology, from the aspect of the SASPP, and you will be contacted in due course by the treasurer, and your cash and certificate will be hopefully in the post soon. So thank you very much, and thank you very much for a very exciting program, a very historic program. I'm sure this is a very fitting way to end the International Year of Plant Health, even though it's somewhat belated now, more than 3 weeks. But thank you again once more. To close.
I will close off. Well, thank you very much. All that remains for me to do is to thank you and the council for your enthusiastic support all the way through for doing this online. I think just looking back on the day, I mean, it really was fantastic. Really so amazing to listen to the science from Sophien's opening to the postdocs and PhDs to what we've just seen in the end now on this award. So thank you to you and the council for your support. Also, thank you again to Sophien and the other speakers for the trouble that they've made to give us this excellent day. Thank you, Tuan and Mornay, for always being there to support and make sure that things run smoothly, and almost for putting a lot of work in to get the talks together and present that for us. So a round of applause to all of you. We will make a recording of this meeting available to SASPP members, so we'll communicate that on the list server for people that some people apparently couldn't have internet problems and so forth, so that would be nice. And ask the speakers whether they would be willing for us to put the talks up as well, so we might spread it even wider. And then I'll end by saying that I really sincerely hope that we see you here between 23 and 26 January next year. So let's all hold funds, and that would be possible to do this in person next year this time. Thank you for joining us, everybody, and I wish you a good day. Good on new people if they want to say goodbye.
I can't do it. They have to do that.
Bye-bye.
Bye, Godfrey.
Thank you. Bye. Bye.
Thanks, everybody.
Bye.
Goodbye.
Bye.
Bye.
Bye from Ida.
Bye, Ida. Oh,